live chat. Live chat, live chat. Done. So hi everyone, welcome to PW NEET English. I'm your biology educator Ambika Sharma, and in today's session we are going to discuss human physiology and plant physiology. We are sorry for starting a little late, some technical glitch was there, but I hope now I'm perfectly audible and visible. Am I? Yes everyone, am I? Hi everyone, am I audible and visible? Perfectly? Sure? So do let me know in the chat section whether I'm perfectly audible or not, because today's session is going to be very amazing, right? And in today's session, we are going to cover the most important, the most expected chapters of your class 11th biology. So ready, excited? How's the energy? Ma'am, cell cycle kab tak hoga? Like, I have one doubt, like why do people always focus on the things which are pending, but why not which we are going to do? है ना? Right? So it's about the mindset, guys. So this is what I genuinely want to advise you, that if you really want to crack this examination, it's very important to have that cool. It's very important to maintain the calmness. Then only you can crack this examination, because cracking the examination is not just the syllabus, how much you know about a topic, what's your level of knowledge. No, it's not like that. How well you plan your day on the exam, how well you start your day on the examination day, plus how well you attempt your paper, right? Are you in a normal state of mind, or are you panicking? All the things, they are going to make the difference, right? Sometimes we panic and we lose the marks. So these are also the kind of things which you need to take care. So rather than ma'am, this chapter is pending, that chapter is pending, you'll get the recorded video for that as well, right? But why can't you focus on the thing, okay, this is what we are going to start today, hai na? Okay, so what is the criteria for today? Today the criteria is very simple. So it's a one-shot lecture where what are we going to target? We are going to target the plant physiology as well as the human physiology. Are we going to target the complete plant and human physiology? You know that we have less time. So today's target is the most important topics from these two units, guys. Most important topics from these two units, right? And they are literally going to be very, very, very important. Okay? Okay? So, what do you want me to start first? Plant physiology or human physiology? What do you want me to start first? Quick. From plant physiology, you know that from plant growth and development, PGRs are important. So we'll be starting with that, right? From photosynthesis, okay, quite important. Here we will discuss all the possible things, your Z scheme, PS, PS1, all the basics we are going to cover, and same for the respiration. So overall, they are very important. You are not allowed to miss anything from that, right? Directly from plant growth and regulators only, beta, 2 questions minimum you can get this time. Imagine, just from one topic only. And I'm always mentioning this thing to you all, and I always want you to focus on this particular thing, that do not miss the examples, do not miss the diagrams and tables from the NCERT. That's what you have to do. So let me see the majority. Human plant, whatsoever you will say, I'll start. But yeah, your energy is quite low. I'm not happy with that, right? I'm not happy with that. I won't take your much time. I'll directly start, but I'm not happy with your energy. What you people are doing? First of all, hit the like button. If you are new to our channel, just quickly subscribe to our channel. In this journey, right, we have to increase the subscriber number as well. So share this channel with your juniors, right? Share this channel with your seniors as well, right? Who are thinking that they'll prepare later or whatsoever it is. Just share this particular channel, because now onwards, right, guys, the amazing content will be there on this channel, right? Will be regular here. You know that already our batches are going to start from 20th. Our batches are also starting. But meanwhile, it's not like that we are not going to take the classes live on YouTube. This time you will see that yes, we will be teaching in our live batches also, but meanwhile we will keep teaching on YouTube as well, right? Right? So this is the amazing plan for all of you. But I am not happy with your performance here. No chat, no energy in the charts, nothing at all in the charts. What you people are doing? right? And I will go with the majority, whatsoever you will say. And I can see plant physiology, plants first, it's short. ठीक है भाई, whatsoever public will say, we'll do that. We'll start the plant physiology. But today I need a promise before starting my class that nobody is going to leave the class today. You have to be with me, because whatsoever topics I'm going to pick, they are going to come in your exam, and with the same energy and same enthusiasm, guys, you have to study. You won't believe, from last 3 days I'm not keeping well, but I know this time is so crucial. I cannot afford to take a leave. Okay, so if I am that energetic, I want my students to be as energetic as I am. So is it a deal? Is it a deal? So are you going to support this channel? Are you people going to support this channel? Are you going to push it? Yes. I don't need any, anything to say here, because the brand name here is PW. And you know that it's a brand, right? And it's not a brand with somebody, it's a brand of students. Okay, it's a brand of students. Because of students, this brand is at this level, right? People here don't need anything, right? We have students' love. That's why this brand is here. So you know that they are student-centric. You know that the platform is amazing. You know that the features there are amazing. So I'm repeating this again. From 20th, we are going to start the batches as well, right? And you will see a different level of energy, consistency, practice there as well. But yes, on the YouTube also, we are not going to compromise. The quality content will be provided to you. If you know me earlier, you know that when it comes to the quality content, I have never compromised it, and I will never. Okay, so let's start the plant physiology, and let's start with a very simple and a very basic chapter, which we call as, Hanji, what is it? Plant growth and development. That's the chapter that we are going to start, people. So what is the name of the chapter? The name of the chapter is plant growth and development. So let's get started. Excited? Okay, so I will start from a very basic point here. First of all, what is the meaning of the word growth? You tell me. What is the meaning of the word growth? Yes, everyone. What do you understand with the word growth here? Like, you are also growing, na? Hai na? Right, I am to an old person. You people are still growing. And in my case also, growth will be there. But how, what kind of growth will be there? Now, it is going to be the aging. It is going to be the senescence. That's what I can say. So, basically, what I want to say is that when we use the word growth, beta, growth is actually an irreversible, right? Growth is actually what? Growth is actually an irreversible, permanent change. Okay. What's that? It is an irreversible, permanent change. That is what growth is. In very simple words, I will start. What is that? It is an irreversible, permanent change, right? And why that change will occur? And why am I saying irreversible? Right? I'll start with the very basics here. The same thing you people also read in the living word. So we are saying that it is an irreversible, permanent change. Like, let's suppose if something is growing, right? It's not like that it will come back to its basic form first. So it is an irreversible, permanent change that occurs due to, definitely due to metabolism, right? So it's an irreversible, permanent change that occurs due to what? That occurs due to metabolism. So beta, what exactly is the metabolism here? So metabolism means the chemical reactions which are going on in our body. What's that? Chemical reactions that are going in our body, that are going in a body. So when we talk about these chemical reactions, now please understand the basics here, right? Because I know plant physiology is difficult for many, so I'm starting from the scratch. So you please pay attention. So when it comes to the metabolism, we have two words. One is anabolism and another is catabolism. And trust me on that, people. Trust me on that. These words are also important. So one is anabolism and another is catabolism. So from cata, what you have to remember from the word cata, you have to remember the word cut. To break something, right? Cut, to break something. So here you will say breakdown. What will you say here? You'll say breakdown. And when I'm using the word anabolism, so it means build up. Now, what I want to specify from this particular part, it's very simple that when I talk about the metabolism, the chemical reaction, either there is the breaking down of something or there is the build up of something. That is how the things are. Breakdown, build up, breakdown, build up, breakdown, build up. And that's how our body is growing. That's how everything is happening. But now, if I need to understand what growth is, I need to understand that if I have to grow, right, the build-up should be more than, anabolism should be more than catabolism. Are you getting my point? Anabolism should be more than catabolism. That is something which should be there in our mind, that anabolism should be high, catabolism should be less. How can you relate it? It is very simple. Now let's suppose if you have to save the money. Yes, beta. Everybody, do answer this question of mine. If you have to save the money, what you are supposed to do? Are you supposed to like you need to save that, na? Like you have to lower down, you have to cut down your expenses. Yes or no? Yes or no? Yes or no? Tell me. Tell me. है ना? So what should be more, expenses or savings? If you have to save money. Quick. Obviously, you have to cut down your expenses. That is what we need. So same is the case. So if we want the growth, so for growth, this need to happen. The anabolism should be more than catabolism. That's what you have to keep in your mind. This is what growth is. So growth is an irreversible permanent change that help in increasing a path, an organ like this. So here the anabolism should be higher than the catabolism. So this is the basic meaning of growth, and these are the basics here. Now my point is, right, when you talk about the growth, right, then I will talk about this particular chapter, right, because I'm relating it with other part also. We have two types of growth, students. One is your internal growth and the another one is your external growth. So this is what you have. You have internal growth as well as external growth. Now when I'm talking about the internal growth, what is the word that you have? It is intussusception, right? That is the meaning of internal growth. So living beings are going to show that. Who are going to show that? Your living beings are going to show that internal growth, that there is an internally, right? We are also growing internally. Our bone length, it is all increasing. So we are growing internally. So when it comes to the external growth, so that is accretion. So when I'm talking about the accretion, A for accumulation. What is this? A for accumulation. Yes or no? Yes or no? So that is what we have. Accumulation is there, right? Why? Because here the deposits will be there, the layers will form. Are you getting my point? The layers will form. So that's how you will see that the growth is. Okay, that's how you will see the growth is. So this is what growth is. Now we are saying plant growth and development. So before starting this part, because many students are expecting me to start from the basic, from the scratch, don't worry. With whatsoever topic I'm going to pick, I will start it from the scratch. So now what you have to understand here, guys, you have to understand one very simple thing. Now the next word is development. Next word is what? Development. So this is what development is. Growth plus differentiation. What is development, people? This is what development is. Growth plus differentiation. So that's why I'm connecting each and everything here. So if I'm saying, I already told you what growth is. So now do you know what differentiation is? Now do you know what differentiation is? Quick. Quick. Guys, quick. So growth plus differentiation is the word. So we already know what growth is. Now we have to understand the differentiation. So as I said, I will start from the basics. So you know basics. So you know that we all develop from what? We all develop from zygote, right? So zygote is exactly what? Zygote is a totipotent cell. What sort of cell it is, guys? It is a totipotent cell. Anybody in the class? Anybody here in the class who can tell me the meaning of totipotency? Anybody? And this line is a direct PYQ, just keep it in your mind. right? So development is equals to growth plus differentiation. Growth you already know. Now we have to reach to the differentiation. So if anybody here in the class is well aware of what differentiation is, do let me know. Do let me know. And guys, the energy should be high here. Okay, the energy should be high. And if you are new to the channel, you know what you have to do. Quickly subscribe the channel. Quick. Yes, what differentiation is? Dekho, first of all, I told you that zygote is a totipotent cell. What is totipotency? So totipotency is the ability. It is the ability of a single cell. It is the ability of a single cell to develop into a complete organism. It's the ability of a single cell to develop into a complete organism. So what is my point here? My point here is very simple, right? From this zygote, even if you talk about the humans, that's how humans are growing. A complete human being will develop from the zygote. So you know that initially it is what? Single cell, then 2-cell blastomere, then 4 will be there, right? Then 8 will be there, and so on, so on, so on. So initially the multicellular structure will form, and with the time what is going to happen, my dear students, right? Every cell, slowly they will start making the layers, and that layers will start making the different parts of the body. For an example, we have already covered that in our previous marathon where we discussed about the germ layers. So you know that when zygote is growing, right? Morula stage, blastula stage, gastrula will also form. Gastrulation occurs, gastrula will form. So when you people talk about that gastrula, in that germ layers are there. So initially it's just a cell who's developing, who's becoming multicellular, then it is forming the layers, the germ layers are there. And when you talk about these germ layers are going to make, what? They are going to make the individual parts, the systems. Ectoderm, it will form our nervous system. Your mesoderm will form our skeletal system, our muscular system. Are you getting my point? Are you getting my point? So this is what you people need to remember. This is what you people need to focus. So why is that taking place? Why is it so? Why is it occurring? What is the requirement of that? Right? So basically, initially your cells, they were just dividing. Right? They were just dividing. So at that time, their fate was not fixed. Are you getting my point? At that time, their fate was not fixed. Their fate was not fixed. They were not clear that what we have to make. They were just simply dividing because all the cells are having the same genetic information. Is that clear? Now with the time, what will happen? With the time, what will happen that cells, in their case, obviously it is because of, cytoplasmic determinants. Yeah, I would say in simple words that it is because of the genes, the proteins that will express the cells, they will get differentiated. So they will get differentiated means they'll become specific, means their specification occurs. Are you getting my point? Their specification occurs. That is what going to happen here. So you will see that initially they are just dividing. Later on, what will happen? They will get differentiated. So when I'm using the word differentiation, means I'm saying that their fate will become specific, right? So their specification occur, they will become specific, that is, they will start having a particular function. So always remember, it is the DM which you have to remember. What you have to remember, guys, it is the DM that you have to remember. So D stands for differentiation and M stands for maturation. Are you getting my point? That's what we need to focus. That's what we need to understand, that D stands for differentiation and M stands for maturation. Are you getting my point? So differentiation as well as maturation will occur. So when a cell gets differentiated means it will be mature now. So initially it was just dividing. And later on what will happen? When its fate will get fixed, then it will become mature. So this is what I have to keep in my mind. Any doubt here? Any doubt here? This is a very important phenomena from plant growth and development, which is equally important for the anatomy as well. Now, little bit I will relate it with the anatomy also. This is what differentiation is, that their fate will get fixed. Obviously, gene expression is going to play the role. Now, there is one more word, that is de-differentiation. There is one more word, that is what? That is de-differentiation. Now, de-differentiation. So, I will use my favorite example here. जैसे let's suppose, है ना? So are you ready for the imagination? Are you ready for the imagination? Yes, but I will also teach in Yaqeen English. But right now let's focus on these 2026 aspirants. They need our support. Hanji. So I'll ask you a question. Just imagine you cracked the NEET examination. Now you are done with your MBBS. You are a MBBS doctor. But after doing this, right, after this MBBS, you have realized, right, you have realized one thing, ki yaar, nahi, I want to, do something else. I want to do something else. This is what you want to, right? Then you started studying again. Are you getting my point? That you started studying again. Okay, so when you got this thing, right, now you are a MBBS doctor, this is like a kind of permanent tag you got, a kind of specific position you got, that you are a MBBS doctor. But now you're like, nahi, I want to study again. I want to do something else. For that, obviously, you have to start studying. Let's suppose you decided that I want to be an IAS officer. So you started preparing for the UPSC. So when you are studying again, means you are again in that phase where you are trying to seek something else, trying to get something else. So this again, once after getting a title, when you again start studying, it means you are again, right, in that dividing phase in terms of plant cells. That's how we understand. Like, let's suppose these cells, right, meristematic cells. So these meristematic cells, what were they doing? These meristematic cells, they were basically dividing. Now they have acquired a permanent function. They became permanent tissue. Now after becoming permanent tissue, they regain the ability to divide, right? They regain the ability to divide. Are you getting my point? They regain the ability to divide. So when they have regained the ability to divide, it means they are de-differentiated. And after that, when again they become or they acquire a permanent fate, then you will call it as redifferentiation. So these are the terms, these are the words that you people need to understand very carefully. So how are we going to start? It is very, very simple here. Firstly, what happened? Just a minute. Okay, so firstly, what happened, guys? The meristematic cells will be there. They will keep on dividing, right? So once they will acquire a permanent fate, means they are differentiated, means they are specified. After that, what are they doing? After that, after becoming permanent, if they will regain the ability to divide, means they are de-differentiated. That is how you people get your secondary meristem, which help in increasing the girth, which help in increasing the diameter. And when, so this is the de-differentiation, right? That again they have started dividing. Then after that, what is happening? After that, again when they acquire a permanent function, just let's suppose, right, you were UPSC aspirant, but you qualified that. Now you are an IAS officer, right? Imagine yourself. Now you are an IAS officer, Aditya. So obviously now you are re means again re-differentiated. So is that clear? Is that clear? Yes. So these are the basics that you need to understand, that are also helpful for what? That are also helpful for understanding the, right? For understanding what? That are also helpful for understanding the anatomy also. So here you can see we have already discussed that what exactly is the growth? Growth is one of the most fundamental and conspicuous, like prominent feature, prominent characteristic feature which we notice. So it is an irreversible permanent change, permanent increase in the size of an organ or in any part. Then we are saying that growth will occur due to the metabolism. I told you already about that, and we know that anabolism need to be high. Anabolism need to be higher than the catabolism if we want the growth. So leaf expansion is also the kind of growth. Now some general basic things, then we will just move to the plant, growth regulators. So plant growth, because here also students, they get confused. So guys, I'm starting this from the scratch, from the basics. 2 questions from PGR, 1 question from this part can also come. So the chapter which you are studying, it's a very short chapter, but it will give you how many marks? It will give you around 12 marks. So ready for that? Hanji, ready for that? Ready? So who's going to hit the like button? Who's going to subscribe the channel? Quick, do it. Chaliye. Next word is the plant growth is indeterminate. What is the meaning of indeterminate? Like here, while reading the chapter, you are going to find the word indeterminate, open, yeah, indeterminate, yeah, closed. These are the kind of terms. Now we have to understand what these terms are. Just see, we know that plants can grow throughout the life. We know it very well. They can grow throughout their life. So this ability is due to the meristematic tissue. Yes or no? They can grow throughout the life. Why is it possible? Yeah, how is it possible? Because of the meristematic tissue, which is at the different location. So this meristematic tissue can divide and it is self-perpetuate. Basically, this meristematic tissue, what is happening here? The cells are dividing. They are also replenish itself. So they are just dividing. So cells basically, they are growing, right? We know that they are in the state of the division. Now, when it comes to the growth, we know that meristematic tissue will keep on adding the cell. It will keep on adding the cell. It will help the plant to grow. Now, new tissue will also form. है ना? New tissue will also form. Now, here the point is, see, read this line very carefully, that growth in plant where new cells are continuously added by meristem and any cell has the potential to divide and form new tissue या organ. That's the keyword here, guys. Please pay attention. Growth in plant is what? Like, we know that due to meristem, continuously new cells will be added. Continuously new cells will be added. This is the first point that I have to remember. The second point that I have to understand here is that because here, right, all the cells, whatsoever new cells are making, they have the potential to develop into any sort of cell is the open form of growth, right? It is the open form of growth. So because growth is continuous, that's why it is indeterminate. And why is it open? Because the new cells that we are making, it's not like that they are fixed. Okay, I will just make parenchyma. Yeah, I will just make sclerenchyma. That cell is like I can develop into anything. So that's why it is the open form of growth. Any doubts? Any doubts? Thank you so much, beta. Any doubts? Any doubts? Sure. So this is the open form of growth. Done. So apical meristem, which is at the tip of the root, at the shoot. So cells are dividing, they are forming new tissues. So this open is the keyword here, guys, because here simply we want to say that whatsoever new cells are forming, they are also, right, these new cells can develop into any other cell. So new structure can arise continuously from these undifferentiated cells. That's why they are open. So indeterminate. So while answering the question in the final paper, make sure that you focus on these words. So what is that? The word here is indeterminate. So it is what? It is indeterminate. It is the continuous is the word here. Continuous. If instead of indeterminate, if they'll mention the word determinate, means that is growth is not continuous, it is fixed. Just in the case of a plant, you know that leaves, they grow only up to a certain size. The fruits, they grow only up to a certain size. So that's how you have to understand this part. So when it is the closed growth, what is happening? Right. First of all, so when it is the closed growth, what is happening? In the open form of growth, the cells, they can develop into anything. But in the closed form of growth, the future fate of cell is already determined. So I'll give you the example again. Let's suppose, right? Have you seen the movie 3 Idiots? Beta, class notes will also be provided in our WhatsApp group. Hanji, have you seen the movie 3 Idiots? Quick, tell me. Three Idiots? Huh? So in that movie, they brought up that concept that, we are after these things, that our kids should become this and that from their childhood. Parents start planning something like that. So now let's suppose if there is a baby, right? In that baby's case, like parents are not at all bothered. They are saying ki theek hai, whatsoever you want to become, you can go for it. There is nothing, right, ki you have to be the doctor only, you have to qualify the NEET only, you have to go for the JEE only. Nah. You want to be something else, you can go for that. You want to be dancer, singer, whatsoever it is, right? You want to be these days, so content creator, right? Or you want to go in the finance, you can do anything. So means there is no bar. Like you want to do anything, you can do anything. Okay. Now, if let's suppose in a family, parents are saying, no, you just have to be a doctor. We are the doctor family. We want you to be the doctor as well. So it means the fate of that baby is already fixed. Even if that baby wants to be something else, no choice. You have to be a doctor. Are you getting my point? You have to be a doctor only. So that's what I'm trying to specify. So when this fate is fixed, ki bhai, even if you don't want to be a doctor, still you have to be a doctor, means closed form of growth. But if they are like, okay, we belong to a doctor family, you want to go in the finance, go for it. You want to go for the J, you go for it. Right? You want to be an actor, just go for it. Open form of growth. So that's how you can relate that. Right? That's how you can relate that. This is how we have to study, and that's how we are going to teach you. Got it? So why closed? Because the meristem is closed in organization with fixed initials producing fixed tissue, means that cells, they'll be clear that this is what we have to make. Okay, no choices, beta, no choices. So differentiation, dedifferentiation, redifferentiation, I have already told you. So yeah, interfascicular cambium, right? Already in the previous marathon in dicot stem, I told you there. So this forms due to the dedifferentiation. Cock cambium, which is also known as phellogen, it forms due to the dedifferentiation, right? So these are the examples, and examples are important. Please note down. Please note down and make sure you sit with your NCERT, because yes, you have to underline the points, you have to revise certain points. Done, beta? Done? Okay. So let's get started. Let's move to the next part. So this is the diagram that I would like to specify here. So you people can see this very clearly. Okay. So shoot apical meristem, it is at the tip here, right? These cells will help in the primary growth. Root apical meristem, though in this diagram they are showing it at the tip, but it is subterminal, right? It is subterminal. What does it mean? What does it mean? It means, see this. See this. In the case of a root section, I hope you all have seen that. There is a root cap, and then after that root cap, right? After that root cap, you will see the meristematic zone. After that root cap, you people see the meristematic zone. Then, I believe you remember there is an elongation zone. I believe then you finally remember that there is the maturation zone. So that's what we study there. Are you getting my point? So meristem is not at the tip. At the tip, there is a root cap which will protect this tip, right? Which will protect this tip when it is growing down. Okay, so root apical meristem is subterminal. That's how you have to decode the diagrams as well. So here you can see the arrows, they are towards this direction. So what are they showing? They are showing the position of the secondary meristem. This secondary meristem, guys, it is also known as lateral / cylindrical meristem. Are you getting my point? So it is the lateral / cylindrical meristem. So it helps in it increases girth. Yeah, it helps in increasing girth, right? It helps in increasing diameter. That's what you have to understand. So this is what they are showing here in the diagram. Diagram is important. Mark it. Separate diagram based sessions will also be there. Just keep it in your mind. So in dicot root, dicot stem, you are going to see that. Next point is very simple. They are saying ki growth is measurable. Yes, you can measure that. Now, in the case of a single cell, when that cell is growing, beta, cytoplasmic growth is there. Directly you cannot measure, but there are some indirect measure. So best way to measure the growth is ki we can check the numbers, but it is not always possible. So what you have to understand here, like increase in protoplasm is difficult to measure directly. It is full-fledged NCERT. Don't worry about that. Increase in protoplasm, it is difficult to measure directly. So what we need to measure, we should measure the quantity which is more or less proportional to it. So growth is directly difficult to measure. In the case of this protoplasm, we cannot measure it directly, right? We cannot measure indirectly. So what we have to do, we have to measure the quantity which is more or less proportional to it. Is that clear? Is that clear? So variety of parameters are there. We go for the fresh weight, dry weight, length, area, volume, cell number. Now, here the point is, जैसे dry weight है, fresh weight है, right? So dry weight will not consider the water component, right? So all that things are there. So growth can be expressed as increase in cell number as well. So yes, if you check the PYQs, you will find the questions from these particular terms. So one single maize root apical meristem. Focus on this thing. One single maize root apical meristem will give you more than 17,500 new cells per hour. That's what you have to remember. Komal, whatsoever is important, I'm already discussing, right? Your focus is on the things which are not discussed, right? So deviate your focus, focus on the points which I'm discussing because they are literally the most important one. So it's a humble request, beta, either answer the questions in the chat section or ask the questions, but please make sure that you ask relevant question at this point of time, because we don't have much time. That's the problem. That's the problem. Right, in the regular session, when we will start our regular session on YT, even in our batch also, there beta, it will start from this, from scratch. Like I'm teaching a KG student, hai na? Right? In their case, I will also tell you, this is how you have to hold the pen. This is how you have to open the NCERT. That is how you have to read the NCERT. That's how you have to underline this word. That is how you have to make the short notes. But there in batch, even whether it is Yakin or it is Lakshya or it is Arjuna, there we all teachers teach you like this. We even teach you how to hold the pen. Okay? Okay. So focus here. So this information is important. So make the short notes. Just write one thing. Increase in cell number in front of that. One single maze is equals to 17,500 new cells. More than 17,000. Now, in the terms of size, what do you have? A watermelon can increase the size up to 3,50,000 times. This is what we have to remember. And in the case of pollen tube beta, you focus on the length. In the case of leaf, you focus on the surface area. So one extra point that you all can write down is, for pollen germination, right? For pollen germination, that's what you have to focus. Boron is very important. Even calcium is important, but the most important is boron. Okay? It's boron. Understood? Boron is very important. Okay? Okay? Done? Chaliye. Next is beta phases of growth. Jaise already I told you, just focus on this part and do not even miss the description from the diagrams. Jaise here they have given ki detection of zones of elongation by the parallel line technique. Zones A, B, C, D immediately behind the apex have elongated most. So this is what, just say, let's suppose what sort of question I can frame from this part. I can frame one sort of question from this part. I can just give you this question, right? And I can ask you which type of method it is, right? By which we are measuring the growth. Yes, right? By which we are measuring the growth. I can simply ask you that. So if you will not read the description, how will you get to know that it is a parallel line method? Yeah, parallel line technique. So that's the beauty beta. Here everything will be covered. Don't worry about that. So now when you are talking about the growth, I already told you, just root mein to root cap is there, then comes the meristematic zone. So beta, when it is the meristematic zone, na, cells are what? Cells are small in size. They are small in size. Whenever you are talking about the dividing cells, right? Whenever you are talking about the dividing cells, so you people can see this particular thing. Okay, you people can see that particular thing. So meristematic cell, they are small in size. So when cells they are dividing, they are quite small in size beta. They will have a conspicuous nucleus, that is a very large nucleus. Surrounding cytoplasm is also going to be dense. Okay, if I talk about the cell wall here, it is going to be primary cell wall. That is going to be thin, that is going to be flexible, that is going to be cellulosic. Are you getting my point? It is going to be thin, flexible and cellulosic. That's what you have to remember. Okay, so this basically is what? These are basically compact. They are basically what? They are compact, they are tightly packed. So here you people will see that among these cells, there are abundant, right? There are abundant plasmodesmatal connections, right? Which are the continuous cytoplasmic connection here. So here you will see that cells are small in size because they are metabolically very, very, very active students. So there will be no stored food here. Vacuoles either are absent or if they are present, they will be of very small size. So these are the points that you have to remember here. Are you getting my point? So these are the meristematic cells. So yes, the question can come from this part. So at the shoot tip and at the root tip. But in the case of shoot, always remember, I'm specifying this again, that meristematic tissue is basically subterminal. What's that? It is basically subterminal. That's what I have to remember. Am I clear? That's what you have to remember. Am I clear? Sure. So cells in this region are what? They are basically rich in protoplasm, guys, right? So already told you they are rich in protoplasm. They have conspicuous, or I would say prominent nucleus, plural is nuclei. Cell wall is thin, primary and flexible, made up of cellulose. Abundant plasmodesmatal connections are there. Anybody having any doubt? Hanji, anybody having any doubt? Anybody is having any doubt, then please ask me. Quick. Quick, everyone. If there is any doubt, do let me know. I'm waiting. Quick. Yes? No? Chaliye, let's go. So, cells proximal, meaning nearby to this meristematic zone, there will be the phase of elongation. So as the word itself is saying, the elongation zone is there. The cells are elongating, their length will increase, and these are the cells which genuinely helps in the primary growth, which genuinely helps in increasing the length, people. So here you people will see the vacuolation will be increased. So that is again a PYQ. Cell enlargement is there, vacuolation will increase, new cell wall deposition will be there. These are the characteristics. So yes, this time one MCQ is expected from this part as well, and it is easily doable portion. Okay, chaliye, next part is, then finally we know that we are having what? Maturation phase. So if it is mature, means cells, they have attained their maximal size, right? Their cell walls are thickened, the protoplasmic modifications are all done and dusted. Basically they have acquired a fate. Is that clear? They have acquired a fate. They know what we have to make or what we have to do. Any doubt? Any doubt here? So that's what you have to keep in your mind. So the next important topic here is the growth rate. So if you guys have noticed that here, I'm not leaving even a single topic. Can you see that? Can you see that? Yes, Dhruv beta. Here, I'm literally not leaving even a single topic. Do you know the reason? Hanji. Do you know the reason? Because genuinely, beta, this chapter is a very easy chapter. And from this chapter, literally, beta, from this chapter, you people can get question in the paper. And this time, and trust me, if you will revise it well now, right, revise it well now, you can score full house from these chapters. Right, you can get full house from these chapters, so pay attention. Now, next is the growth rate, hai na? Next is the growth rate. So I'll give you a very simple example. Just we all say, oh, we are studying, we are studying, we are studying, we are studying. This is what you people say, ki you are studying. Now, let's suppose we have students from the chat. Ha, Dhruv. Dhruv is saying ki, madam, I have completed na 2 chapters in 1 hour. In my division, I have completed 2 chapters in 1 hour. Now, Shravani is saying, ma'am, I have completed 2 chapters in 3 hours. है ना? I can compare. I can compare, yes or no? I can compare easily. कि अच्छा, one is completing 2 chapters in 1 hour and you are taking 2 hours or 3 hours. So they both are studying, but now I will compare their rate. Who's faster, who's slower? It is like that, right? That's what you are supposed to remember, that when you are talking about the growth rate, so growth rate is increased growth per unit time. What's that? It is the increased growth per unit time. What's that, people? Increased growth per unit time is the growth rate. So the rate of growth we can measure mathematically as well. So here you people have 2 words which you have to remember. So make sure you take the screenshot for that as well. So I am writing it here. So growth rate, and here I have arithmetic growth rate, and this side I have geometric growth rate. Arithmetic growth rate is there, geometric growth rate is there. Now in this case, what is the scenario? 1, 2, 3, 4, 5, like this. Please keep it in your mind. This side it is going to be, let's suppose there is 1 cell, then 2, then 4, then 8, then 16, then 32, so on. So basically, what am I trying to tell you? We have 1 cell, that 1 cell will divide into 2. Okay? Out of these 2 cells, this cell is saying कि I am not interested in dividing further. Matlab, I have 2 cells here from this 1 cell. This cell is saying कि I am not interested in dividing further. Right? I will just mature, I will just grow. This one is like, nah, I will to divide. So, what will happen? This cell will stay as such. Yes or no? This cell will stay as such. Yes or no? And what about the cell here? This cell will be like, chalo koi baat nahi. It is okay. I will divide like this. From one cell, you got two cells. Now this cell is just further, it will just mature, right? It will just further mature, and this another cell will be like, no, I will divide. Now when it is dividing, right, it is again forming two cells. So here also one cell is saying, I will just mature. I will not divide. This cell will be like, okay, fine, koi baat nahi, I will divide. So again the same story. Again the same story. Now you can see one cell, two cells, three cells, four cells. How come four? Because this one is also growing, this one is also growing, and this one will be like, chalo, we will divide. So how many cells you got here? Four cells. That's what you have to remember, arithmetic growth. Now, in the case of geometric, which we also see in the starting of that zygote, when zygote is growing, two cells will form. These two are like, theek hai ji, chalo, let's divide. Let's make four. And these four are like, okay, let's divide. Let's make it 8. Then these 8 will be like, let's divide, let's make the 16. So that is the difference. That is the difference here. Is that clear? Is that clear? So this diagram is very important, showing the zygote dividing, geometric phase, all the cells are dividing, then cells capable of division. This, they are dividing. Other cells have lost the capacity to divide. This is again a previous year question, my dear people. So the very first thing that we have to understand here is the arithmetic growth rate. So are you people ready? Quick. Are you people ready? Quick. Arithmetic growth rate. Are you people ready? Sure? Done? So you can see that here we will see that after the mitotic division, only one daughter cell will keep on dividing, other will differentiate and mature. So that's why I have given you this trick that it is DM, first differentiation, then maturation will occur. So it is DM, first differentiation and then maturation will occur. Is that clear? Is that clear? So simplest expression of arithmetic growth is root elongating at a constant rate. So here the expression is going to be beta LT is equals to L naught plus RT. So you should know what R is, you should know what T is. So LT means length at time T, L naught length at time 0, R is the growth rate to the elongation per unit time. What is this R? Do mention it. Maybe it will be a CYQ. So it is the growth rate to the elongation per unit time. Am I clear? Am I clear? Sure, people? Sure, people? Done? Sure? Chali. So here you people can see this thing very clearly. So R is the growth rate to the elongation per unit time. Is that clear? So R is the growth rate to the elongation per unit time. So here I would like to specify one thing. Here you people can see that the x-axis is showing the time, y-axis is showing the height of the plant. X-axis is showing the time and y-axis is showing the height of the plant. So now guys, this is the origin, right? In this diagram we are showing the growth rates. How can we consider it at 0? So it will not be at the origin, it is going to be like this. It is going to be like this. So these are the minor details, beta, which we have to focus. Otherwise, there will be a problem. There will be a problem. Genuinely, there will be a problem. Let's suppose what kind of question I can frame from this part. Simple. I can give you 4 diagrams in the paper. I can give you 4 diagrams in the paper, and if I will give you these 4 diagrams in the paper, right, so I can ask you which one is showing the arithmetic growth rate, your root elongation at a constant rate. So you people should know. Let's suppose everything is fine. Might be in some diagrams here they have mentioned height, here they have mentioned the time, might be instead of this they can mention something else. And let's suppose if they have mentioned time and height here, but if this line is from this part, this is also incorrect. So be very specific, everybody. Be very specific, everybody. So that is how, right, you have to take it. So this is what you people have to focus, right? Are you getting my point? right? So it is not at the 0, it is not at the origin. Kindly take care of that. Kindly take care of that. So it is a constant linear growth where there is a plot of length against time. Is that clear? Is that clear? So then is the geometric growth, which I already told you. So it is this, right? S phase. Again, it is not at 0. So here in this particular growth, what will I see? Here in this particular growth, what will I see? This is the S-shaped growth. This is the sigmoid growth curve. This one is the sigmoid growth curve. So initially there is the lag phase where the growth is very slow. Then it will rise exponentially, exponential phase. So you know that there will be limited nutrients and everything. Then finally there comes what? There comes the stationary phase. What will be there? There comes the stationary phase. Am I clear? Am I clear? There will be the stationary phase. So that's what you have to remember. So this is your geometric growth. So here you can see the initial growth is going to be slow. It increases thereafter at an exponential rate, right? So you know that both the cells can divide here. So because the nutrient supply is limited, so after some time the growth is going to slow down, and then there is the stationary phase. So this is a sigmoid curve. This is the S-shaped curve. Is that clear? S-shaped curve. Is that clear? So sigmoid curve is the characteristic of living organism. Do underline this particular line. Done? Do underline this particular line. Am I clear here? Everybody, am I clear here? Sure? So sigmoid growth curve is the characteristic feature of living organism. Those who are living in the natural environment, this is what they are going to have. So the expression is W1 is equals to W0 ERT. So yeah, there is a PYQ, what E is. Even in ecology, you study that. It is base of natural logarithm. W1 is the final size. So here you also consider the initial size, and obviously initial size will decide that final also, right? So what is the R here? Just here. What we said for the R. Wait, let me show you. Growth rate to the elongation per unit time. What we just said there, growth rate to the elongation per unit time. Beta, it is the absolute growth rate. Just again, I will give you the example, Muskan. Again, I will give you the example, Muskan, Shravani. Let's suppose I am saying कि यार Muskan is studying, है ना? Muskan has completed 5 chapters in a day. Okay. Now I'm saying यार Shravani is also studying. She has also completed 7 chapters a day. Now I'm talking about their individual growths, right? In the case of Muskan as well as in the case of Shravani. So what is that? That is basically the absolute growth rate. Now if I start comparing, right, that they were at this point, in that much time they have done this, in that much time they have done that, then I have started comparing. Matlab relatives. Relatives do what? Relatives, their job is to compare. So these are what relatives are. Yes or no? What the relative job is, just to compare. So that's what you have to remember. So R, there R was the absolute growth rate, that is growth rate to the elongation per unit time. So when R is the relative growth rate, it is the measure of ability of the plant to produce new plant material. Matlab, it is also the efficiency index. Right, what is the efficiency of that plant? Because if it has formed the new plant material at a certain rate, it is telling me about their efficiency. So here final size depends on the initial size. When this is what I'm considering, beta, I am talking about the relative growth rate. Am I clear? Am I clear? So this is the one that I'm going to explain to you. Okay, so this is the one that I'm going to explain to you. Ready? Ready? Sure? Sure, everyone? Are you all ready? Sure? Done. So here you people can see these are 2 leaves. In this particular leaf was at 5 centimeter square. Matlab its initial growth, if I have to say, is initially it was at 5. Finally, it reached at 10 centimeter square. So in general, if I'll say what is the growth rate here, I know absolute growth rate is 5 centimeter square. Now, if I talk about this particular leaves as well, 50 centimeter square reached at 55 centimeter square. Right, again here also the growth is 5 centimeter square. But what we have to focus, we have to focus on one thing, that from where it started, from 5 to 10, mane kind of double. It was at 50, then it had just reached to the 55. So growth is more here, beta. So sometimes now we compare ourselves with others, right? I think it's very common with you all, right? You might compare your results with others, right? You might think that, oh my God, why that student is getting 660 marks? Why am I getting just 500 only? So we have this basic comparison in our mind. Sometimes we are like, yo, I just have this bicycle. That person is having this car or this bike or something like that, right? So beta, we always need to see one thing from where we started. Right? Sometimes we start from the scratch. Sometimes we don't have that ancestral wealth. With time, we develop everything. Right? With time, we work hard. We earn money. We gain that status. We work hard for that. Right? And some people, they born with that status. They have that status ki yes, they are already in a kind of rich family. They are already in a kind of reputed family. So when you do such comparison, na, just think about one thing. We always need to focus on one thing, that from where we started. Are you getting my point? Some people start from here and then they reach here. Some people start from here, and even if they are reaching here, means that's a growth, and one day they will also reach here. So that's how you should see your life as well. Okay, so sometimes while learning these things, we learn a lot of things as well. Like, it's not just for the syllabus, these lessons are also for the life. Now come back here. So these are the two leaves. So here you can see, when I'm saying this is the quantitative comparison between the growth of living systems. So there are two ways for that. So when there is the measurement and comparison of total growth per unit time, right? The first thing is what? The first thing is ki measurement and comparison of beta total growth per unit time, right? That what exactly is the total growth? Here also 5 centimeter square, here also 5 centimeter square. So growth of given system per unit time, this part is basically absolute growth rate. Growth of given system per unit time, this is actually the absolute growth rate expressed on a common basis. Example, per unit initial parameter is the relative growth rate. So in relative growth rate, you'll see the absolute growth rate, but you also have to consider the initial parameter. That is the most important thing. Is that clear? That is the most important thing. So two leaves are given here. So here you people can see the relative growth rate is equals to growth of the given system per unit time to the initial parameter into 100. Right? Growth of the given system per unit time to the initial parameter into 100. Is that clear? Is that clear? So one leaf A, 5 centimeter square reached to 10. B was at 50, reached to 55. So when I will see the growth rate here, so for the relative growth rate, I will consider absolute growth rate to the initially. So initially one leaf was at 5. Multiply by 100. So 100% is the growth rate. So initially the absolute growth rate is 5. Initially other leaf was at 50. Multiply by 100. So you will get 10%. So that's how you compare it also, right? That's how you have to answer these things. Any doubt? Any doubt? Guys, so where is your energy? Where is your Josh? So I want to see some fire emojis in the chat section, and be fast, quick. Be fast, quick. Quick people. Done? So if you have seen my strategy video, there also I posted that there is a table which you have to remember. There is a table which you are not supposed to leave. That is that table. So here you people can clearly see we are talking about the development. So development means all the changes that are taking place in a body, in an organism. Right? During our life cycle, whatsoever changes are taking place in our case, right? Or that complete is the development. Now here, because we are talking about the plant, so you people will see, right? You people will see that from that seed germination to the senescence, everything is the development. This table is very important, again from the NCERT. So you have a meristematic cell. So it is showing plasmatic growth. Plasmatic growth means it is showing increase in, I would say, protoplasm ya fir cytoplasm, you can write. And I believe you know the difference in protoplasm and the cytoplasm. Protoplasm, first living substance, right, including cytoplasm plus genetic material. Cytoplasm, you already know, the main arena of cellular activities. That's what you have to remember. Okay, got that? Got that? So meristematic cell, plasmatic growth is there. Then there will be the expansion or elongation. Then finally there is the differentiation and maturation. So first it's D, then it's M. Finally the mature cell will undergo aging and the death will occur. So this point is clear. Quickly tell me, this point is clear? Yes, please. Done, beta? Any doubts? Sure? Understood? So that's important. So that's what you have to remember in your mind. So next is the plasticity, beta. So what exactly is the plasticity? Basically, how living body is responding to the environmental changes. Are you getting my point? How living body is responding to the environmental changes. That is what plasticity is, right? So here you can see that plants, they follow different pathways. How come they follow different pathways? In response to environment or the phases of life. Are you getting my point? Plants, what these plants are doing, these plants, they follow the different pathways. How come? In response to the environment or to the phases of life to form what? To form a different kind of structure. So these responses are plasticity. So plasticity means what? Please pay attention. Different pathways in response to the environment or the phases of life. So they are the different pathways in response to the environment or phases of life. Just heterophily is the example of the plasticity. Hetero means, what is the meaning of hetero? Hetero means different. Hetero means what? Hetero means different. Phily means leaves, right? So different types of leaves are forming during the lifespan. It can be because of the, during the lifespan, yeah, because of the habitat as well. I'll give you an example. Just say we have the cotton. So cotton scientific name is Gossypium. Coriander is there, Coriander sativum, larkspur is there, and even we also discuss the example of ranunculus, right? Ranunculus, which is buttercup. So basically, if you talk about the coriander, in the case of coriander, you will see that the juvenile plant and the adult will have different shapes of leaves. They will have different shapes of leaves. Now, we have the plants in which you will see the, if let's suppose this is the plant, if the part of the plant which is under water, here you will see dissected leaves, right? And which one is above the water? Here you will see complete leaves. So that's what you people need to remember. So yeah, development is important. It is controlled by the internal external factors both, right beta? So heterophily is the example of plasticity. So examples here are very important. Just a larkspur is given, right? So you should know their scientific name. Buttercup, ranunculus is also given. You should know their scientific name. So water habitat may you have the dissected leaf, and in the terrestrial habitat, these are the leaves that you have. So please keep it in your mind. Is that clear? Please keep it in your mind. Is that clear? So finally, the most awaited topic, plant growth regulators, where here again, I'll give you the keywords. So ready for this? Ready for this? So we just took 1 hour and we are done with this other part of part. Now we will take another, 30-40 minutes, and then here we will be done with the plant growth and regulators as well. So I hope up to this part, everything is clear. I hope up to this part, everything is clear. Yes. Sure? Sure? Pakka? So ready for PGRs? So PGRs overall are important. Stick to the NCERT. Today also I will teach you the part from the NCERT only. So I hope you are ready. Anji, sure? Okay. So phytohormones, or we are saying PGRs, plant growth regulators. Okay? This is the word that we have. First of all, pay attention here, beta. The word is regulators. What is the word? What is the word? What is the word regulators? To regulate something. When we are saying we are regulating something, we are not simply saying that we are suppressing it, right? No. To regulate means as per the requirement. When there is the requirement to promote something, it will be done. When there is a requirement to inhibit something, that will also be done. This is the meaning of the regulation. So regulation doesn't mean, right? Regulation, it doesn't mean ki you just have to increase something or you just have to decrease something. Now, as per the need, increase or decrease has to be done. So we are talking about the plant growth regulators, PGRs. So here we have two categories. One is the plant growth promoters. Another is the plant growth inhibitors. Are you getting my point? One is the plant growth promoters. Another is the plant growth inhibitors. So in the promoter category, you have auxin, you have cytokinin, you have gibberellin. Are you getting my point? Are you getting my point? Auxin, cytokinin, gibberellin. In inhibitors, you have ethylene and then you have abscisic acid, which is also known as ABA, and it is also the stress hormone. So what's the main point here? That your ethylene, right, can come in both the category. Ethylene can act like both, right? Can act like both promoter as well as inhibitor. Promoter as well as inhibitor, but majorly an inhibitor, right? That's how you have to remember this. Majorly an inhibitor. Are you getting my point? So they will promote the growth, right? They will help in the growth. Jaise auxin, natural forms mein we have IAA also, indole 3-acetic acid. Cytokinin, it is a kinetin, right? It is basically also called kinetin, but kinetin is obviously not the natural cytokinin. The natural one is zeatin. So it is basically a purine derivative, that is N6-purpuryl aminopurine. Then gibberellin is also there, gibberellic acid beta. That is again we obtained that from the terpenes, right? Then you have abscisic acid from the carotenoids. Ethylene is there, which is obtained from the amino acid methionine, which is a sulfur containing amino acid. So all that points are also important, but that's what you have to remember, right? So let's target it one by one. Okay, let's target it one by one. So both the categories are there. So we will talk about their discoveries. But for their discoveries, the one thing that you have to remember is that for all the, right, that for all 5 PGRs, their discovery is serendipity. Their discovery is serendipity. Do you understand the meaning of serendipity? Serendipity means by chance discovery. What's the meaning of serendipity? It means by chance discovery. Is that clear? Is that clear? What is the meaning of that? What is the meaning of that? It is the by chance discovery. Is that clear? By chance discovery. Okay? So that's what you have to remember. So yeah, here I'll suggest you to stick to the NCERT, right? Because here some extra points are also there. But yeah, I want you people to stick to the NCERT because of this time constraint wala thing. Our exam is there, right? And within few days, so we have to make sure to get best out of it. So rather than just showing you that, oh my God, this is what I know, this is how much I know for a topic. Now, this is not the right time. Right at this point of time, I just need to make sure that whatsoever I teach, whosoever is following my lectures, they should get best out of that. Okay? Okay, so here you people can see the Auxin discovery. Firstly, we have to discuss. So the scientist name, the scientist contribution is very important. So it is the observation of Charles Darwin and Francis Darwin, right, his son. So actually they were not looking for the auxin. They were actually studying the phototropism, right? That how, you know, these plants and all, they show the bending towards the light. This is what they were studying, but they found what? Ultimately a chemical. So you know that when there is a unilateral source of light, can you see that? The bending, right? The bending is there. The bending is there for what? Towards the light. So basically when you talk about the Charles Darwin and his son, they basically studied the canary grass, right? They basically colutiles. Mane, you can simply keep it in your mind, right? You can simply keep it in your mind. And what is that? Colutiles, the tip of the canary grass, right? Right? Okay, so in the comment section, I want you people to help me out here. You will tell me what is the scientific name of canary grass. I don't know, right? So help me out in the comment section. Later after the class, you will put that. That's your homework. I want to see your response as well. And guys, see here there is a poll. If done, right, do let me know. So tell me, did you smash the like button or not? And not just the like button. Who's going to subscribe the channel? You have to subscribe the channel as well. So just focus there. So we have the canary grass. So you are providing one source of light there. We'll see that they will grow towards that. Yeah, bending is there. This is the phototropism. So series of experiments were there. Actually, they observed this thing that they are showing the bending. So they were like, there is something, right? Charles Darwin and his son Francis Darwin is like, there is something here at the tip, right? There is something which is forming at the tip and that something is going downwards. That's why from one side there is extra growth and the bending is occurring. So further after that, another scientist was there, right? If Boynton Jensen. So he was like that something is a kind of mobile, like they said a mobile signal is there, like a movable signal is there, which will be there here at the tip. And from the tip, it is going downward and it is actually guiding, right? It is actually guiding that, okay, it has to show the bending. Is that clear? That it is actually guiding that it has to show what? It has to show the bending. Is that clear? Is that clear? Yes or no? Right, that it has to show what? It has to show the bending here. Are you getting my point? Now, after that, what is the next point that we have to see? Yeah, we have to study here. Now, the Boynton Jensen, when they studied, they were like that mobile signal, that movable signal which is formed at the tip and going downward. It is nothing, but it is a kind of chemical signal only. And later on, right, because this is what we have in the NCERT, F.W. Wendt, he was the one who extracted that from the tips of the oat seedling. So oat means scientific name is Avena, right? Avena species. So it is from this is what they have extracted. So is that clear? Is that clear, everybody? So any doubt from this? Any doubt from this? Yes. Yes. Are you sure? So this is what you have to remember about the auxin. So I'll give you some extra and the important point here. For auxin formation, you need the amino acid tryptophan, and auxin is the plant growth regulator which shows the polar transport. So I'll explain you the meaning of that also, but make sure in your NCERT you write these 2 points. Quick. And guys, if you are very good, Aarti has answered my question that, ma'am, canary grass is Phalaris canariensis. Excellent, beta, excellent. Too good. Yes, please. Yes, please. Quick. So let's go, let's discuss that. So that's what you have to remember. Now, what is my next point here? I said auxin, tryptophan, this amino acid, the most complex amino acid, is required for its formation. And I told you that it shows the polar transport. It means auxin, it doesn't move, right? It doesn't move randomly. When we are saying that polar transport is there, so it means it doesn't move. It doesn't move randomly, right? Rather, it doesn't move randomly, rather it shows a directional movement. So your auxin, it forms in the root as well as in the shoot tip, right? So in shoot, right, we will see its movement is beta basipetal. Basipetal movement is there. Auxin will show what? Auxin will show basipetal movement. That is from tip towards base. Right, towards base it will show. In the case of root also, beta, you will see, right, the movement, matlab it can occur in both the direction, but generally the movement is there, right? Generally the movement is there in a proper direction there as well. So mainly you will see downward in central tissue, upward in upward layers in the case of root. ठीक है बेटा? Okay. Any doubts? Any doubts? Sure. So main direction is in shoot, that is basipetal. Okay, so root mein to beta I told you, right? In roots it is a complex pattern, downward in central tissue and upward in that side one. So basically it depends on the, and gravity also contributes there. Next, my favorite hormone, plant growth hormone here, gibberellic acid. So if the word is acid, mane acidic in nature. So here basically what is about, so Irichi Kurosawa in 1926, right? Matlab their detailed discoveries are very interesting, but here we have to stick to the NCERT. So gibberellic acid. So like we have the growth hormone, na, from this gibberellin word you have to remember G. G mane growth. Growth. Again, growth is there. What is there? Growth is there. Guys, make sure to subscribe the channel as well. So now we are talking about the gibberellic acid, right? Now we are talking about the gibberellic acid. So gibberellic acid, its discovery was also very interesting. So in Japan, you know, farmers, they were noticing that something was happening to their rice fields. They were observing that rice seedling, they were growing excessively and then they were falling down. Right, they were growing very excessively and then they were just falling down, right? So Irichi Kurosawa in 1926, years are important, they reported the appearance of symptoms of disease in rice seedling, right? So when they were treated with sterile filtrates of fungus. So basically what happened is Japanese farmers, they noticed that their rice seedlings are growing excessively, right? And then they were falling down. It's just being foolish that you are growing excessively and falling down. So this is the Bacan is the word that they use for it. Bacan, might be it is having some different pronunciation, but yeah, Bacan, foolish seedling disease. So they observed that, right? The scientists, they observed that it is caused by a fungal pathogen, right? So what is the name of the fungi? Gibberella fusaricorae. Gibberella fusaricorae. Now its name is fusariform. Now its name is changed, Gibberella fusaricorae. That time we used to call it, now it is fusarium, right? So basically Gibberella fusaricorae. So Kurosawa, he means Ichi Kurosawa, he observed that not just that fungus directly, even if we have a culture where we have grown that fungus, if we use that medium, right, in which fungus is not there, if we use that filtrate directly, in that filtrate there is no fungus, we are using that filtrate only, even if we use that filtrate, that is also causing some disease. So then they got to know that might be fungus is making a chemical and that chemical is causing the disease. Right, that might be fungus is forming a chemical which is causing that disease. So initially they were thinking that it is just limited to the fungi only. Then they got to know that plants also produce it naturally. Right? Thank you, Beta Raja. So plants, they form it naturally. So that's what you have to remember. That's what you have to keep in your mind. Now, next is F. Skoog and his co-workers. This is again my favorite discovery. Why? They were working on the tobacco callus culture. Just see, they from the internodal segments of tobacco stem, the callus, a mass of undifferentiated cell proliferated. So I'll tell you, just they were doing some tissue culture in the lab, right? Just let's suppose they use the tobacco plant. So when we do the tissue culturing, we take the part of that particular plant, we call that plant as part as X plant. You can take anything here. Then beta, you give some hormones, right? Hormones, carbon source, like food and everything you provide, because you want this X plant, because here they all are permanent, right? This is the permanent tissue. We want it to become meristematic again. So when you provide all that chemicals, all that hormones, so this X plant will form a mass-like structure. Right, this X plant will form a mass-like structure, a cell mass is there. So this is an undifferentiated cell mass. This is an undifferentiated cell mass. What's that? It is an undifferentiated cell mass. That's what you have to keep in your mind. Undifferentiated cell mass. So it is just dividing it is not having a fixed fate. This is what callus is. So they were trying to see, because initially they were thinking ki yaar auxin can help, right? But later on when they were doing that tissue culturing, they realized ki nahi yaar, it is not just the auxin, right? It is not just the auxin that can help here. They were like, there are some other chemicals. So they were trying many things. They were not getting the results. So sometimes it happens na, that out of frustration, we are like, we throw something. This is what they did, right? This is what they did. Literally, this is what they did. Matlab, there was an old bottle in the lab. There was a chemical there. They were just like, and then they noticed, yeah, growth is there. Then they realized ki yaar, what was there in that particular chemical, like what was there? Then it was having the DNA. They tried to put the DNA, but they didn't get the result. Then they realized ki that was an old DNA. So might be that is, destructed or something like that. And then finally, they got to know that. But this is basically, firstly, they got the chemically synthesized, right? This cytokinin. Cyto means cell, kinin, division. It induces the cell division. So that a synthetic one was kinetin, but the natural one is zeatin, which they obtained from maize, basically, right? They obtained that. Is that clear? Is that clear? So that's what you have to remember. So in addition to auxin, nutrient medium, they were supplemented with extracts of vascular tissue, yeast extract, coconut milk, or the DNA. So Miller et al. Means Miller and colleagues in 1955 identified and crystallized the cytokinesis promoting active substance that they termed kinetin. So what is the keyword here? What is my keyword here? That when I'm talking about kinetin, what is my keyword here, guys? That when I am talking about kinetin, it is not natural. This one is synthetic. This one is not natural. This one is synthetic. Am I clear? Am I clear? Any doubt? Guys, quick. Now start with your doubt so that we can go ahead. Quick. Hanji, doubt. Doubt. Done? Sure? Yes? Thank you, Jaya. Got that? Okay, chaliye, let's go ahead. So the next discovery is during mid-1960s. During mid-1960s, three independent researchers, right? During mid-1960s, three independent researchers, right? They reported the purification and chemical characterization of three different kind of inhibitors. They were working independently. They were not aware of each other. They were like, okay, you are like, they independently in different corners of the world, they were working, they were experimenting, they found some chemicals, right? They named it accordingly, but later on when their chemical nature was checked, so we got to know they are all same. So what was that? Just say one gave it the name inhibitor B, another is abscission 2, another is dormin. So later on it was named as abscisic acid, plant stress hormone. So next is the HH Cousins 1910. Year is also very important. Match the column kind of question can come from the years. What sort of question can come from the years? Match the column. So they confirm the release of volatile substance from the ripened oranges that can speed up the ripening of unripened bananas. So the fruits which were ripening, they were releasing a chemical. It was a gaseous hormone, right? It was the gaseous hormone beta, so which was increasing the ripening of the unripened bananas. Okay, just sometimes you might have noticed that if you have, the fruit basket, if one fruit is over ripened, it can destroy others as well. Why? On what basis? How can they do so? How can they do so? It's simple, because of this ethylene. So this is about the discovery. So it is the auxin which was discovered first. If somebody will ask you which one was the first plant growth regulator discovered. First plant growth regulator discovered. Which one helps in the fruit ripening? Your ethylene. In the case of ethylene, I have to, and ethylene is the gaseous plant growth regulator here. So ethylene, its bioassay is triple response. Whenever you hear the word triple response associated with ethylene, now I will explain that to you. Okay? 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 Sure? So should I start? Okay. Okay, beta. So here you can see auxin, indole compounds. So you have IA, that is indole 3 acetic acid. IBA is also there, indole 3 butyric acid. They are natural, and synthetic one is naphthalene acetic acid, 2,4-dichlorophenolic acetic acid. This is a very important question, and here I would like to specify IBA can come in both. It can come in both natural as well as synthetic auxins. Natural as well as synthetic auxins. Is that clear? Am I clear here? Are you sure? Am I clear? Sure, guys? Sure? Any doubts? Any doubts? Pakka? Okay. Chaliye. So let's get started. So cytokinin told you adenine derivative N6-furfuryl aminopurine. Gibberellin terpenes will make that. Ethylene, it's a gaseous hormone. So methionine amino acid, which is a sulfur containing amino acid, is very important for it. Abscisic acid, carotenoid derivatives. Yeah, this is something that you should know. This is something that you should know. So ethylene के लिए aqueous solution में it is ethiphen, which is CEPA as well chemically. I'll discuss that also. So take the screenshot. I'll talk about it then. Quick. Take the screenshot, then I'll talk about it, guys. Quick. Everybody quick. Sure. So take the screenshot, though I will share the notes in the Telegram group also. So you can see that, right? They say auxin, first also isolated from human sperm. FW went, isolated it from tips of oat seedling. The discovery, the accidental hormones also. So kinetin, not naturally in plants. It is found from the autoclaved herring sperm of DNA. Zeatin is the natural cytokinin. So you people can clearly see. I have added the tables as well. So my students can easily revise that, right? So these are basically for your quick revision. Right, now I will go back to the NCERT part, right? So they are basically for your quick revision. So are you liking the notes as well? Are you liking the notes as well? Ki Hanji madam, we are liking the notes. They are literally going to help us to revise quickly. Sure? Are you sure, people? Are you sure? Pakka? Arey yaar, your energy is quite low. You are demotivating me. First time it is going to happen. Students are demotivating a teacher. है ना? Isn't it? मतलब I think first time this is going to happen कि students are demotivating a teacher यार. Very bad, very bad. Very bad. Thanks Komal. But what about your energy? I'm not liking it yaar. Nah. No energy, nothing at all. Just sitting like this. Just chit-chatting. Nah, बिल्कुल नहीं. Be focused, participate here in the discussion. Okay, participate here in the discussion. Sure? Paka? So should I continue? So you like the notes? You like the notes? Ki, ma'am, that table is good. So that's how you have to make the short notes. Don't worry. I joined this platform late, right? But for NEET 2027 aspirants now, I'll be like, ki, just I'm going to hold your ears like that, and I'm going to rotate it even through the online classes. Okay, even through the online classes, I'll tell you, I'll teach you how to make the short notes as well. We'll show you in the live lectures how to make a copy, how to make the notes, what to write, what not to write, right? And might be this time I will just give you my handwritten notes only, right? And I'll obviously you have to make your own as well, but we'll tell you that, idiots, that's how you have to make the notes. That's how you have to write everything. These are the main pointers that you have to focus, right? In the platform, there is a kind of Pitara box also, where you just have to click. There will be the NCERT paragraph. Just click on there, right? Just click on there. Just get the questions on the basis of a particular paragraph of NCERT. Okay. So that's the student-centric approach, and that attracted me to join this platform. So for NEET 2026, I'm giving the most important one. For NEET 2027 from day one, right? Once we will start our YouTube series, na, we will give you that things also, the lectures also. Okay. But YouTube पर हाँ MCQ practice and timing वाला thing is little difficult. Otherwise तो you will get everything. So focus here. आ जाओ, come back. So auxins, from Greek, auxin means to grow. Okay. So it was first isolated from human urine, my PYQ. Okay. The term auxin is indole 3-acetic acid. So this is the most occurring natural auxin. There is another natural auxin also, right? Your IBA. For IBA, do remember this, right? IBA stands for indole 3-butyric acid. It stands for indole 3-butyric acid. This is your IBA. Indole 3-butyric acid. This is what your IBA is. So it can be both. It can be both natural as well as synthetic auxin. Natural as well as synthetic auxin. Why synthetic? Because yeah, right, it is also synthesized in the lab also. It is used again in the horticulture department. Yes, beta. So right. So this is what you people have to remember. Thank you, Shravani. So indole 3-butyric acid can be both natural as well as the synthetic auxin. It can be there in both the categories. So when they are saying synthetic compounds, yes, you have the example of NAA, you have the example of 2,4-D, even you have the example of 2,4,5-T as well. Let me elaborate that. Please highlight it in your NCERT, guys. Kindly highlight it. So NAA, naphthalene acetic acid, guys, it is the synthetic auxin, right? Not formed naturally, but yeah, in the lab. So 2,4-D, D is dichlorophenoxyacetic acid. So 2,4,5-T is basically your 2,4,5-trichlorophenoxyacetic acid. What's that? It is the trichlorophenoxyacetic acid. Right, it is the trichlorophenoxyacetic acid. Right, this is what we have. Done, beta? Done, beta? So that's what you have to remember. Okay, so they are generally produced by growing apices of the stems and root, from where they migrate to the regions of their action. So I told you, auxin will show you what? Polar transport. They'll show you what? Polar transport. That's what you have to keep in your mind. Okay. So all these auxins, they are used extensively in agriculture and horticulture practices. So they are helpful to initiate rooting in stem cuttings, another important topic. So this is what I would like to highlight here, guys. This is what I would like to highlight here, that for rooting in stem cutting, this is what they said, na? What do you need? You need Mr. Auxin. So basically, beta, not just auxin, you need auxin and cytokinin ka ratio. Cytokinin ka ratio. Just let's suppose if there is, this is very important for organogenesis, high auxin, low cytokinin, high auxin, low cytokinin, what will happen? Rooting. If there is high cytokinin and there is low auxin, done, there will be the shooting. Is that clear? There will be the shooting there that you have to remember. Okay, so this is how we use it. So auxin promote flowering in pineapples as well. So this is very important, right? Not in all, but this specific examples are something which we cannot leave. But auxins ke case mein, the very important thing is the apical dominance. What is the meaning of apical dominance? It is its main function. Just let's suppose here in the tip region, we know that the auxin formation is there. So because there is a tip, it will not allow the lateral buds to grow, right? Due to the presence of this tip, this is the apical dominance, right? Hormone is also going down that. So it will not allow the lateral buds to grow. So if we decapitate it, if we remove the tip, decapitate it means remove tip. When you remove the tip, then the lateral buds will grow. So this is what apical dominance is, that the presence of apical bud, right? What's that? That the presence of apical bud will not allow the growth of lateral buds. Is that clear? Is that clear? Sure? Are you sure? Yes, everybody. Are you sure? So apical dominance is very important, and overall you know that it's a promoter, so it will help to prevent fruit and leaf drop at early stages. Matlab at early stages, at immature stages it will not allow, but if there is a mature leaf, right, the mature fruit, then it will be like you fall down, so that we can utilize your nutrients and we can give it to the growing one. Very important. So as I told you already about the apical dominance, so this is very important PYQ beta. So shoot tip decapitation, matlab when we remove the shoot tip, it can result in the growth of lateral buds. Is that clear? So it is very important for plantation, right? It is very important for plantation and for the hedge making. Is that clear? Is that clear? So due to the apical dominance, what will happen? Plant will grow taller than bushy due to that. Is that clear? Any doubts? So it is because of the auxin gradient, high auxin concentration near the apex, lower towards the base. So that's why the lateral bud will remain inhibited, because high will be there at the tip, at the base it will be lower, so lateral buds will not grow. That's what you have to remember. So it can also it is used in this tea plantation and hedge making. So it also induces parthenocarpy. It will do what? It induces parthenocarpy. Parthenocarpy means formation of seedless fruit. Matlab without formation, without fertilization, fruit formation will occur. Without fertilization, fruit formation is occurring. So you know that seed is the result of what? Seed is the result of fertilization. So without that it is taking place. एसी नहीं चल रहा, कोई आइडिया? एसी नहीं, वो वर्किंग नहीं है, चल नहीं रहा अभी जी. Done. Thank you. Huh. Okay, beta. So that's what you have to remember, okay? So now in tomatoes for parthenocarpy it is helping. Now it is widely used as a herbicide. Herbicide, it will kill the herbs. So 2,4-D, again it is a PYQ. It's a very important PYQ. So 2,4-D is the word dichlorophenoxyacetic acid. So it will basically kill the dicot weeds. Is that clear? It is going to do what? It is going to kill the dicot weeds. It is not affecting the monocots, but will kill the dicot weeds. So for weed-free lawns, we can use that. So yes, even it is having a very important role here in the tissue culturing as well. So as I said, as of now, just stick to the NCERT, right? Do not go beyond NCERT. This is very, very important for you. Now the next is gibberellin. Always remember it like this, G for growth hormone. And here, the main thing that you have to remember is bolting effect, and even gibberellin can basically replace vernalization. I'll tell you what is this. Understood? Chaliye, let's go back. Chalo, aajo. Let's start. So gibberellin, as I said, is a growth hormone, and again, gibberellin, terpenes are used for their formation. So stick to the NCERT. So there are more than 100 reported gibberellins, right, from the widely different organisms such as fungi and higher plants, right, such as fungi and higher plants. So they are GA1, GA2, GA3, right. So gibberellic acid was one of the first gibberellins to be discovered, beta, right? So all the gibberellins are acidic. Do underline that. Gibberellic acid, that's why the name is na. So I'll tell you what bolting is. Just we have the rosette plants. Just there are some plants na, in which you will see so many leaves are there, like your cabbage and all. So many leaves are there. Actually, they have the internodes, but they are quite short. Like for an example, if I'm taking this example, this one is the node. This one is also the node. And in between, what do you have? You people have internode, right? You people have internode here. Now, let's suppose if this internode length is going to be very short. Internode is the place where the branches, branches, leaves, all that grows, na. So if it is very short, so it will look like this. You are having so many leafy kind of structure together. So when we apply the gibberellic acid, it helps in, it gives this bolting effect. What is this bolting effect? So in that bolting effect, na, prior to flowering, before flowering, the internode length will elongate. Internode length will elongate. That's what you have to remember. Am I clear? That's what you have to remember. Just keep it in your mind, guys. So as I said, so they produce a wide range of physiological responses in the plants. Number one, their ability to increase, to cause an increase in length is used to increase the length of grape stalks as well. So these are the points which you should definitely not miss, especially your gibberellin and ethylene, right? Please be focused there. So gibberellin also help to elongate and improve the shape of fruits like apple. Then they delay senescence, they delay the aging process, but that delaying senescence, which is Richmond-Lang effect, it is the main role of cytokinin. So yeah, gibberellin is also promoting it. Just we know that hormones, they work in a synergy, they work together, right? One hormone helps in another. That is how the things are. So that's what I really want you people to remember. Is that clear? Is that clear? So thus the fruits can be left on the tree longer. So just to extend the market period. So GA3 is used to speed up the malting process in the brewing industry. So GA3 is used to speed up the malting process in the brewing industry. Is that clear? In the brewing industry. So sugarcane, they stores carbohydrates as sugar in their stem. Is that clear? Sugarcane, you know that from that we get the sugar. So when we spray this gibberellin, right? When we spray this gibberellin, it increases the length of the stem. It increases the yield by 200 tons per acre, right? By 200 tons per acre. So that's what you have to keep in your mind. So sugarcane store carbohydrates as sugar. Just keep it in your mind there, beta. So even on the juvenile conifers, if we spray that, it will speed up their maturity period. So early seed production will be there. And this point, this complete point is of bolting, the most important function. So do not miss their most important functions at least. Okay. Do not miss their most important functions at least. Am I clear? 20 tons. Beta 20 tons per acre. I'm not saying 200. 20 tons per acre. Okay. Sure. So stick to the NCERT here. So, just now I told you that it can also replace the cold treatment. Cold treatment, for that we use the word vernalization. Sometimes you know that some plants, when they grow, they also need a cold season, winter season, where the low temperature is given to them. Then they can cause the flowering. है ना? Now, if in such type of species, जैसे some wheat varieties are also there. So in such type of plant species, yeah, in such type of varieties, I would say, you can do what? Those which need the this winter treatment, right? This cold treatment, low temperature treatment. In their case, if you will provide them the gibberellic acid, it will help. So gibberellic acid is overall help in growing. Even if there is a genetically dwarf plants, they can also grow because of the action of gibberellin. Is that clear? Because of the action of gibberellin. Is that clear? Is that clear? So cytokinin है अपना अगला. So this is again very important. So cytokinin N6-furfuryl है ना? N6-furfuryl aminopurine. N6-furfuryl aminopurine, adenine derivative it is. N6-furfuryl aminopurine. Is that clear? So cytokinin में kinetin is there. It's not natural. Zeatin is there, which is natural. Direct PYQ. It is obtained from zymase. And here there are many functions of the cytokinin as well. This is cell division. Cell division is very important, and it shows Richmond-Lang effect. What does it show? Richmond-Lang effect, which means delaying senescence. Right? It delay senescence. So that is what you have to remember. Am I clear? Am I clear? Done, beta. So cytokinins have specific effect on cytokinesis. Cytokinesis is your division, right? You know that karyokinesis, cytokinesis, cytoplasmic division. So they were discovered as kinetin. So they were discovered as kinetin. What does it mean? A modified form of adenine, a purine. Right, a modified form of adenine. So how is it? From the autoclaved herring sperm DNA. So kinetin doesn't occur naturally in the plants, right? So search for natural substances with cytokinin-like activities led to the isolation of zeatin from the corn kernels and the coconut milk. So that's what you have to remember, right? That's what you have to remember. Is that clear? Is that clear? So since the discovery of zeatin, right, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. So natural cytokinins, where will you see? Where the cell division is fast, जैसे root tips में, shoot tips में, even in the developing shoot buds, beta, young fruits, they all are having the cytokinin. So they will form, obviously cell growth is there. That's why, beta, they are delaying the senescence. Are you getting my point? Because it is having the cell growth, so that's why it is delaying the senescence. Are you getting my point? Are you getting my point? So it helps to produce new leaves, chloroplast in leaves, lateral shoot growth, and even the adventitious shoot formation. So cytokinin help overcome the apical dominance as well. So this is very important. Your Richmond-Lang effect is very important. So it's a counteract of apical dominance. So they help in promoting the neutralizing mobilization of the nutrients. This is how you have to remember. So any doubts? Yes, everyone. Any doubts? Any doubts? Beta, Avik, there will be 4 marathons. If you follow them all, definitely minimum 320 you will get if you don't make the mistake. Hanji, doubts? People doubt. Sure, should I go ahead? So yes, cytokinin, I also told you, high cytokinin, low auxin will help in the shooting. है ना? High cytokinin, low auxin. Right? High cytokinin, low auxin. ठीक है? Shooting. High auxin, low cytokinin, rooting. That's important. ठीक है? Should I go ahead? So ethylene in the food ripening, right? Ethylene, where do we need it? Ethylene is required for the fruit ripening. Is that clear? Is that clear? Done? Done? Sure? Any doubts? Hanji, everybody, any doubts? Okay, so ethylene. In the case of ethylene, you have to remember SEPA. You have to remember triple response very clearly. Okay? So let me tell you what exactly they are. Done? Ethylene is a simple gaseous hormone. It is synthesized in large amounts by tissue undergoing senescence, right? Those who are undergoing aging, right? Those which are undergoing aging, there you will see their formation, just ripened fruits and all. Okay? It is a gaseous hormone. You have to remember it. Its discovery is also very interesting. Just you know, in very early, na, जैसे coal lamps were used. So when coal lamps were used, so people observed that the plants which are living nearby to, which are growing nearby to them, they were showing a very different kind of growth pattern. Like in their case, their axis, it was swelling, right? Their axis, it was swelling. The stem, they were showing the thickening. Kind of hook formation was also there in their case. These were the kind of observations, basically. So people used to think that it is because of the coal gas, because initially it was believed that it is only a pollutant. With time, further when they studied, they got to know that ethylene is actually also made by the plant. So when I'm saying the word triple response here, it is also the bioassay of ethylene. It is also what? It is also the bioassay of ethylene. Bioassay means basically when you have to check the potency of a particular chemical, right? In a living organism, how is it working? What is its potency, concentration, and how is it working? Then you talk about the bioassay. So triple response. Just say what is happening? Just say, let's suppose there is a seed. It is trying to germinate. There is a kind of hurdle. So when, just normally we know that, right? It will grow, the stem axis will grow vertically, but instead it will grow horizontally. It will grow horizontally. Yes or no? It will grow horizontally. So horizontally it will grow, the swelling will be there. And then because it has to, it is basically kind of, protecting itself from this hurdle as well. So swelling of axis will be there. Instead of growing vertically, it will grow horizontally. And then there is the apical hook formation. So we will see swelling of stem axis, swelling of stem axis, right? Swelling of stem axis will be there, or swelling of stem axis will be there. Horizontal growth, we will see horizontal growth of the seedling, swelling of stem axis, and the apical hook formation is there, right? Apical hook formation is there. Apical hook formation is there. So this is the triple response. So wherever you will see these three things, we are talking about the ethylene. So here you people can see ethylene on plants include horizontal growth of seedlings, so swelling of the axis, and the apical hook formation in dicot seedling. So this is basically the triple response. So we are saying na horizontal growth of that seedling because there is a hurdle there. So it is also protecting its meristem there. So horizontally it will grow instead of growing vertically. The stem axis is swelling. The apical hook formation is there. So all these points, they are showing what? Triple response, which is also the bioassay of ethylene. So bioassay means when you are using, when you are checking the potency of a chemical in a living organism, bioassay is the word that you are using. So ethylene, I told you it can come in both the categories. So because it promotes senescence and abscission of the plant organs, especially of leaves and flower. So obviously senescence, aging, abscission means they will fall down from that particular plant. So this is why we are saying that their roles are both promoter and inhibitor. So fruit ripening, it is helping. So it enhances the respiration rate during ripening of the fruit. So such fruits where the respiration rate increases, we know that they are also known as climactic fruit. Jैसे your apple, your banana, etc., etc. So you will be amazed to know there is one very amazing question which is given in beta NCERT exemplar. Yes, there is a very amazing question which is given in NCERT exemplar. You might have noticed that, and if not, I don't know why, right? Means you don't study, right? So my point is, जैसे have you noticed this thing that when we go to buy the apples from a shop or somewhere, from a stall or a shop, so you might have noticed that in that, that apple is covered with a kind of paper, kind of wax a paper. Yes. Hmm. Yes, they're covered like that. Noticed? Notice it ever? है ना? Do you know why? Because that covering around it will try ki, because that covering is to make sure that oxygen should not enter there. Now you will be like, why? Because if more oxygen will enter there, the respiratory rate of that fruit will increase, and when the respiratory rate of that fruit will increase, its ripening will be too fast, right? And this is not what we want, because apple is also a climactic fruit. The respiration rate will be fast. So who's enhancing this ethylene? So oxygen will not be there, the respiratory rate will not enhance. So that is how one way or another way it is helping. And I don't know whether you guys have seen it or not. It is given in your NCERT exemplar. It is given in your NCERT exemplar. Okay, so now its promoter activity, it breaks seed and bud dormancy. It breaks seed and bud dormancy. It helps in the germination in peanut seed. So examples where they have given now, they are very important. So sprouting of potato tuber is also done by it. So ethylene also promote internode या petiole elongation in deep water rice plant. From each and every line you people will find the question. So it leaves upper part of the shoot to remain above water. So it promote the root growth and root hair formation, helping the plant to increase their absorption surface. Is that clear? Is that clear? So now do relate it with the ethylene. Say auxin. Say auxin, I told you na, promote flowering in pineapple. Promote flowering in pineapple. Told you already. So ethylene is also used to initiate flowering and synchronizing fruit set in pineapple. So that is, or these are the points which you need to remember. Okay, that is, or these are the points which you need to remember. Okay, so it also induces flowering in mango. So since ethylene regulates so many physiological processes, it's a very important in agriculture. So source of ethylene, note that ethylene is not, ethylene means not ethylene. Matlab ethylene means ethylene, it's just the source of ethylene, right? From where the ethylene can be obtained. So it is the source of ethylene. Am I clear? Source of ethylene it is. So ethylene in an aqueous solution is readily absorbed and transported within the plant and release ethylene slowly. So this ethylene, chemically beta, it is your SEPA, 2-chloroethyl phosphonic acid. Told you already na, it is SEPA, 2-chloroethyl phosphonic acid. Am I clear? 2-chloroethyl phosphonic acid, that's what I told you, right? So it is also helpful for fruit ripening in tomatoes, which we also parthenocarpy auxin. So don't confuse this. As we just read, in the auxin it will induces the it causes the parthenocarpy in tomato, but here it is saying fruit ripening in tomato. So in the paper there is a difference of these minor things. Might be they are asking which hormone will cause fruit ripening in tomato. Then what should be the answer, everybody quick? Then what should be the answer, everybody quick? Then what should be the answer? Quick. Yes. Yes. Tell me, who will cause the fruit ripening in tomato? That is ethylene. But if they are asking, who's causing parthenocarpy in tomato? Then what should be the answer? Do remember, do answer it. So in apple and the tomato, they will do that. It accelerate abscission in flowers and fruit. It speed up the abscission in flowers as well as in fruit. So thinning of cotton, cherry, walnut. So it promote female flowers in cucumbers and thereby increasing the yield. Any doubts? Any doubts? Quick. Any doubts? Quick. Sure. Okay, done. Last is stress hormone, abscisic acid. So being a stress hormone doesn't mean that it gives stress to the plant. No, beta, no. It means it will help that plant to survive in the stressful condition. Plants can survive in the stressful condition because of what? Because of this particular hormone. Just say, let's suppose today your parents are not in a good mood. Okay? Your parents are not in the good mood. Okay? You went home, your sister is telling you ki, see, your parents are not in the good mood, don't say this and that thing, otherwise you will be punished. है ना? She's guiding you, because see, if you will do some things, some absurd stuff, definitely they are going to beat you. So she is like telling you, don't do this, no need to say anything to them. So she is helping you, है ना? She is protecting you in that condition. So same is the case with the abscisic hormone. Seeds, it is telling the seeds ki, see, your conditions are not favorable for you, right? Conditions are not favorable for you. Right now, the environmental temperature condition, whatsoever it is not in your favor. So if you will try to grow, you will not survive. So that is being stupid if you will try to grow. So it is helping it to survive under unfavorable condition. So it's the stress hormone. So not in a bad sense. Don't see this hormone in a bad sense, right? See it in a positive sense. It is helping it to not to challenge such, unrealistic conditions where you won't be able to survive. Just be dormant, be in that dormant state and just take care of yourself, right? So it is also referred to as the anti-transparent hormone. Anti-transparent hormone. Why beta? Why beta? Because when plant is under the water stress, right? When plant is under the water stress, anti-transparent. So when plant is under the water stress, right, under the water stress, so it will make sure that no wastage of water should be there in the form of vapors through that stomata, that is transpiration should not occur. So it stops that. So it is anti-transparent as well. So always remember, it is a antagonist of GA. So what is the meaning of antagonist? So antagonist mane opposite. So it works opposite to your gibberellic acid. Okay. So abscisic acid, you know that it plays a role in abscission and dormancy, right? It is also having other roles also. So you know that it will inhibit the plant metabolism, right? It is going to inhibit the plant metabolism. So it inhibits the seed germination as well. It stimulates the closure of stomata and increase the tolerance of plant to various kind of stresses. Is that clear? Is that clear? So that's why it is the stress hormone. So it is having its role. It is having its role in seed development, in maturation and in dormancy. In seed development, maturation and dormancy. So by inducing dormancy, ABA helps seeds to withstand desiccation and other factors which are unfavorable for the growth. So in most situations, ABA acts as antagonist to GA. Am I clear? ABA acts as antagonist to GA. Is that clear? Is that clear? So this is what we have to remember. So yeah, hormones, they can complement each other function. They can be antagonistic as well, means opposite to each other, जैसे abscisic acid and gibberellic acid, and they can be individualistic. Like individually they are acting on something and sometimes they need another hormone, right? And they can act together. So that is what you have to remember. So flowcharts are already provided to you. So this is all about this particular chapter, right? These short notes will also be shared with you all. So this is the summary part is already done. So let's get back to photosynthesis. Hi. Why this slide? So, strange. Yes. Now, doubts. Time is for the doubts. Any doubts? I don't think that you need a break. We don't need any break, है ना? Or you need a 5 minutes ka break. Do you? I don't think so. Lunch. Acha. But beta, my lunch is not prepared. Who's going to help me here? But ठीक है, so it's 120, है ना? मतलब it's 1:15, so let's meet at 1:30. Done? Let's meet at 1:30. Done? Done. Okay, beta, doubt is, beta, Komal, think about it once. Think about it once. Pay attention. Use this break for the brainstorming. Studio person. मुझे break के लिए वो करना था. So there will be a break for 15 minutes, not more than that. So see you all after that. Am I audible? Guys, am I audible? Do let me know. Okay, so let's start the next part. Let's start the photosynthesis, right? Let's start let me change this as well. Hmm. One second, please. Okay, so it should be black, but I don't know. Okay, guys, so let's utilize this part only. So let's get started. So yeah, any doubts so far? Quick. Any doubts so far? Everybody. So should we start? So yes, which topics from the photosynthesis are difficult for you? Quickly tell me. Quickly tell me. Which topics from the photosynthesis are difficult for you? Yes? सर मेरी क्लास चल रही है, एक बार आप स्टूडियो में आइए, मुझे हेल्प चाहिए. वहाँ पे बाहर तो कोई नहीं है और मैं ऑब्वियसली क्लास छोड़ के नहीं देखूंगी. आप प्लीज़ एक बार आइए प्लीज़. स्लाइड का कलर ब्लैक आ रहा है, उसको ब्लैक नहीं आ रहा है, उसको एक बार जो चेंज कीजिए प्लीज़. Okay, so it will be done. So let's talk about the photosynthesis. Okay, let's talk about the photosynthesis. Let's discuss that. So here in the photosynthesis part, right, beta? So we will go with the very selected part. First of all, I'll tell you what exactly is the photosynthesis, and that you know from class, I believe, 6th. But here, what is the most important thing that we have to take care? In the case of photosynthesis, we know that there are two reactions. Light reaction is there as well as the dark reaction. So we have to make sure, right, that like when we study that light reaction, dark reaction, their location, what exactly is happening there, how is it further proceeding, we need to take care of everything, right? We need to take care of everything. That's what we have to remember. So are you ready for that? So here we will discuss PS1, PS2, what exactly are they. We have to discuss what is the Z scheme. I will tell you the flow. If you have studied that before, then again, if not, even if you are a fresher, you will get to know about it. Is that clear? Is that clear? So let's talk about that. So ready? Ready? Quick. Ready? Yes, everybody show some excitement and energy in the chat section from this particular chapter. Yes, you people will get 3 MCQ in your NEET 2026. So now next 2 hours, right? Next 2 hours are going to be for, right? Next 2 hours are going to be for the Next 2 hours are going to be for what? Next 2 hours are going to be for the 12 marks again. And let's try to reduce the duration as well. Done? सर नई स्लाइड ना ग्रीन जा रही है, मुझे ब्लैक चाहिए उसका बैकग्राउंड. Okay, guys, so we are ready. Okay. Chalo, so let's start. Ready? Fine. So what are we talking? We are talking about the photosynthesis. So we will start with the most important part today. Yes, ready? So we are going to start with the most important part today. Now, beta, when we talk about the photosynthesis, what exactly is the meaning of photosynthesis? You know that the word is a synthesis, means the word is a formation. You have to make something. So what sort of process it is going to be? Is it anabolic or is it catabolic? You tell me. What sort of process it is? Is it catabolic or anabolic? What sort of process it is? Yes, everybody, quick. Quick. So build-up is there, formation is there. So definitely it is going to be an anabolic process. And if it is anabolic, it will use the energy. It is going to be endergonic. That's what we have to remember. It is going to be endergonic. That is what we have to remember. Am I clear? Am I clear? Am I clear? Sure? Done? Sure. So, photosynthesis. In the presence of light, there is the formation of food, and we know that who's going to do that? Your plants are going to do that. So, your plants, what exactly are they? Plants are the transducers. Why are they transducers, students? Because transducers, so they will convert, right? They will convert light energy into chemical energy. They will convert light energy into chemical energy. So that's what we have here. So this is what we have in the case of transducers. So they will convert light energy into the chemical energy. That's why plants are also the transducers. So we know that they'll make the food. They are autotrophic. We all are dependent on the plants for our food. Am I clear? Am I clear? So I believe this part is clear. So what plants need? They need the sunlight, they need the chlorophyll, they need the carbon dioxide, they need the water, they need the enzymes for their process. So overall, what exactly is happening in the photosynthesis? Because firstly, I'm giving you the overview. So basically, photosynthesis is oxidation reduction. You can say that it's a redox reaction. So why it is a redox reaction? Because carbon dioxide is getting reduced. Carbon dioxide is getting reduced and your H2O, right, it is getting oxidized, right? So this is what we have. So reduction of carbon dioxide will make the sugar, will give us the sugar. That's what we have to remember. So overall in the redox react, or overall in the photosynthesis, let me remind you this thing, that if you think that all the photosynthetic organisms, they will always release the oxygen. No, it is not like that, right? There was an experiment was there, right, by Cornelius van Nijl, who said this, so that whatsoever is the oxidizable compound, that will decide, right, that will decide that what exactly will liberate. If instead of H2O, if there will be H2S, then there will be no oxygen. It is the sulfur that will come out, right? So because here at the place of, let's suppose A can be anything. If it is H2A, so it is the A that has to come out. So if water is used during the photosynthesis, then beta oxygen will come out. Am I clear? Then beta oxygen will come out. This is what you people need to keep in your mind. I hope it is clear to all of you. So just have a look of photosynthesis here, so you can see that it's a physicochemical or photobiochemical process. What sort of process it is? It is physicochemical or photobiochemical process it is, beta. And here what is happening, your carbohydrates are synthesized from these inorganic water and CO2 in the presence of light and pigments. Any doubt here? I am pretty sure there is no doubt. So here you can see 6 carbon dioxide, 12 H2O. The reaction has to be balanced. Sunlight, chlorophyll are required. This is glucose, and then water will also form and the oxygen will be released. So why do I call it as the redox reaction? Because I know that water is the oxidizable compound here. Its oxidation will occur during the light reaction, and carbon dioxide reduction will occur during the dark reaction. So in these two steps we study that. So it is basically anabolic. So it is going to absorb the energy. Any doubt here? Any single doubt here? Ki ma'am, we are not able to understand that. Yes, everybody. Any single doubt here? Sure. So this is the basics of, these are the basics of photosynthesis which you all need to remember. We will not discuss the experiments today. This is what we have to keep in our mind. So the next topic that we are going to discuss here is ki what it's both the processes where the light reaction and dark reaction will occur. But before going forward here, I would like to explain you the structure of the chloroplast, because the structure of the chloroplast will give me a better idea, right, about the I would say the locations where this process will occur. Is that clear? Is that clear? Are you sure? Are you sure? Okay, guys. Okay, so let's get started. So yes, if somebody will ask you that is your this what is your photosynthesis anabolic or catabolic? What will you say? What will you say? Is it anabolic or catabolic? What are you going to say? What are you going to say? Yes, everyone. Catabolic or anabolic? It is anabolic. It is catabolic or anabolic? It is anabolic. Chaliye. So now let's understand the chloroplast. So we know that we have different types of plastids. So the most common one is the chloroplast here. So when you talk about the chloroplast, it's a double membrane bound organelle. What sort of organelle it is? It's a double membrane bound organelle. That's what you need to remember. Number one, there is outer membrane, inner membrane. You all can see that, beta. You all can see that there is the outer membrane, there is the inner membrane. In between these two, you people will find intermembrane space. You people will find intermembrane space, which is also known as periplastidal space. Please pay attention. Diagram here is very important, right? Diagram here is very important. So it is the periplastidal space. Now here the matrix here is known as what? The matrix here is known as stroma, right? Stroma, in which dark reaction occurs. In which dark reaction occurs. Are you getting my point? So it is the matrix here in which what is going to occur, So this is the matrix that will occur here. Is that clear? It is the matrix that will occur here. Now, here you will see some membrane bound sac-like structure. But a diagram of chloroplast is very important to understand the location of the various processes here, and they might ask you this only, that where the link, this light reaction will occur, where the dark reaction will occur. Question like this can also come here. Is that clear? So yes, you have matrix, you have stroma, dark reaction will occur here. Now look at these structures. Please look at these structures. So now when you are talking about these structures, so you can clearly see that, you can clearly visualize that, right? See, this membrane bound sac-like structure, beta. This membrane bound sac-like structure, can you see that? This one, what are you calling it? You are calling it as thylakoid. What are you going to call it? You are going to call it as the thylakoid. Is that clear? Thylakoid. So many thylakoids, when they pile up, so what will you get? You will get this structure. This is known as granum. So granum is a singular, right? Granum here is what? Granum here is singular, beta, and the grana is going to be plural. So how will they form when these sac-like structure, I would say membrane bound sac-like structure is basically, right? Membrane bound sac-like structure. Membrane bound sac-like structure. This is what we have to remember. Is that clear? Is that clear? Any doubts? Yes, everyone. Any doubts? Pakka? So this is granum, plural is grana. Now in between these two, right? In between these two, here you have intergranal thylakoid, which is also known as fret lamella, which is also known as stroma thylakoid, right? Because I have to teach you PS1, PS2, so I really need to tell you which part is what. So is that clear? Baki plastids also, you know that they are the this chloroplast is what? This is the semi-autonomous organelle, right? It is not totally dependent on nucleus for all its protein. We know it very well that it is having its own circular DNA. It is having its own circular DNA and it is having the 70S ribosome as well. Am I clear? Am I clear? So that's why it is a semi-autonomous organelle. So I believe up to this part, everything is clear to you. Right? I believe up to this part, everything is clear to you. Now, look at the next part. See this. Will tell you what part it is. This diagram. Firstly, have a look. Then let's discuss that. Quick. Quick. Okay. So now you can see this. This is thylakoid, 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 thylakoid. So they will stack over each other. They will stack over each other. They are forming the granum. And as I said, thylakoid is a membrane bound structure. So in the case of thylakoid, just we study the plasma membrane, that is what we have. So it is beta your thylakoid membrane. Please remember this. Please underline this as well. Right. It is your thylakoid membrane. It is your thylakoid membrane in which light reaction will occur. What will occur there? The light reaction will occur. Is that clear? Where the light reaction will occur. Is that clear? So that's what you have to remember. Right? So now when you look at this particular structure, so you people can see this. You people can see that. Can you see that, beta? Nobody will explain you that depth, but this will tell you where your PS1 is, where your PS2 is. So please pay attention, right? So now when you look at this, we have two words here. One is the appressed region. Another one is the non-appressed region. One is the appressed region. Another one is the non-appressed region. So what is that exposed and non-exposed part? The one which is exposed and the one which is not exposed. So that's what you have to keep in your mind, guys. So you people can see that. So this one is not exposed. It is non-appressed. This one, right? Appressed portion, or I would say appressed portion is also known as tightly stacked. What do you call it? You also call it as tightly stacked. Is that clear? You also call it as tightly stacked. Is that clear? Is that clear? Sure? Sure? So this part, so here you people will see PS1. Okay, so PS1 is non-appressed. PS1 is present in the non-appressed part. Your PS2, which is your photosystem 2, it is in this appressed part. Meanwhile, in the stroma lamella also, you people are having, right, in the stroma lamella also, you people will see that you will just have PS1. Firstly, tell me the locations clear. Firstly, tell me the locations clear. Done, beta? Sure? Are you sure? So that's what you have to remember. The location is very important. So again, you people can see this is basically non-appressed part, exposed part. So in exposed part, what do we have? Exposed part may we have your, right beta? So exposed part may we have your PS1, photosystem 1. And in this stacked part here, we have your PS2 here. And even in the stroma lamella, fret lamella, intergranal thylakoid, this is having different names. Stroma lamella, right? It is having fret lamella, right? Stroma lamella is there. Fret lamella is there, right? Even it is also known as intergranal thylakoid, right? Intergranal thylakoid. So here also you people will see that there is the PS1 which is present. The location, as I said, is very important. Any doubts? Any doubts? So this is the diagram again. Once again, you people can see that diagrams, right? Once again, you people can have a look of this particular diagram, because now our whole discussion will be around it. Ready, beta? Now our complete discussion will be around it. So yes, if somebody will ask you, because we are sticking to the most important topic, where will you see the dark reaction? Where are you going to see the dark reaction, everybody? You will see the dark reaction in the matrix. Where will you see the light reaction? You will see in the thylakoid membrane. So two reactions we have in the photosynthesis. Guys, is there any issue with the stream? Yes, is there any issue with the stream? I hope I'm perfectly audible. I hope I'm perfectly audible. बीच में बफर कर रहा है क्या? वो अभी बफर कर रहा है स्क्रीन पे, अभी देखिए आप एक बार. Yes, everyone. Clear? Done? Okay. So is it buffering or am I visible, audible? Is it still buffering or am I audible and visible? Is there any lag? Now is it fine? Now is it fine? आप यहीं पे रुको, क्लास ऑब्जर्व करो और एक बार इन्हें बोल दीजिए बाहर कि आवाज ना करे बहुत ज्यादा, प्लीज. Done? So is that okay? Is that fine? Sure? Now there is no lag. Chaliye, come back to the session. So where will the light reaction will occur and the dark reaction will occur? So I believe now everything is clear to you. Right, beta? Right? Okay? Sure? Chaliye. So let's discuss it. So what are we going to discuss? We will talk about the photosynthesis. In the photosynthesis, there is the light reaction, which is also known as Hill reaction, right, beta? And there is dark reaction, which is also known as biochemical reaction. This one is also known as your photochemical reaction. Now, what is the meaning here? So let me elaborate this part. So please pay attention. I know many of you are not having the strong basics here. So please pay attention, right? The light reaction and the Hill reaction. Okay, the light reaction and the Hill reaction. This is what we have. Next is the dark reaction and the biochemical reaction. So light reaction, where is it taking place? I told you already, it is in the thylakoid membrane. Where do you find it? It is in the thylakoid membrane where you are going to find it. This is the number one point. It is in the thylakoid membrane where you people are going to see this. This is the number one point. This is the light reaction, or we are also calling it as the Hill reaction. This is the first thing. Now, in the case of light reaction, it is better directly dependent on the light. Okay? So, products of light reaction are what? What are the products of light reaction? Yes, please. What are the products of light reaction. So you will see ATP will be there, NADPH will be there, oxygen will release. This is what you are going to get from the dark reaction. Location clear. What will you get? Is it clear? Again, these are the repeated PYQs. These are the repeated PYQs. Is that clear? Now we went to the dark reaction. Its location is stroma. Its location is stroma. Is that clear? Is that clear? Now, here what will happen? Word dark here is misnomer. Word dark here is what? It is misnomer term. What is the meaning of being a misnomer term? Misnomer term means it doesn't have the same meaning, right? It doesn't have the same meaning what you are getting from it, this particular word. Just for an example, you are sitting, you are living in a particular place. Nearby to you, there is a restaurant which is saying China Gate. It doesn't mean that that restaurant will lead you to the China, right? It is something misnomer. So dark word here is misnomer term here. Why is it misnomer? Because it doesn't mean that this particular process will occur in the darkness. It is actually light independent. It is actually light independent, right? It doesn't depend on light directly. So it stops if the products of light reaction, are not available. Am I clear? If the products of light reaction are not available, it stops if the products of light reaction are not available. So dark reaction needs, right? It needs ATP, it needs NADPH2. Okay, so it is nicotine amide adenine dinucleotide phosphate. That is what NADPH is. Is that clear? That is what NADPH is. So this is what we need. Now these 2 points clear? These 2 things clear? Everybody. These 2 points clear? These 2 things clear? Sure? Sure. Done, beta? So that's what light reaction is, and that's what you have to keep in your mind. Now, before going forward, right, before understanding the basic phenomena, we need to understand that what are the pigments and what are the things that we need here. Are you ready? Yes, everybody. Are you ready? So let's talk about the action absorption spectrum and then let's lead to this particular part. So we will start with the pigments first. Okay, we will start with the pigments first, so pay attention. And chloroplast structure is already done and dusted. One more thing is given in the NCERT, that is the alignment of chloroplast. If you read NCERT carefully, you will find that something is written there. They didn't explain it, but yeah, this is from the NCERT, the alignment of chloroplast. So it is not like that chloroplasts are always going to be at the same position and the same way. Not at all. Their alignment, it changes as per the light intensity. If light intensity is very high, you can see that diagram. If light intensity is high, this is going to be parastrophe. So what is the position of chloroplast? So you can see that light is falling like this. So can I say that chloroplast is arranged parallelly in that case? Yes or no? Do take the screenshot here. Can I say that they are arranged parallelly here? Yes or no? Yes or no? Can I say that? Obviously. If there is the low light intensity, because now chloroplasts are like, we need the light, then it is epistrophe. So here you can see chloroplasts, they are arranged perpendicularly. They are arranged perpendicularly. Then you have moderate light and chloroplast distribution is random here. Right? The chloroplast distribution is random here. So high light may parallelly, low light may perpendicular, in the moderate light random. So these are the technical words for them as well. If you can remember technical words, very good. If not, at least remember high light intensity means parallel, low means perpendicular, and moderate means random. Is that clear? Is that clear? Right? Sure. So this is something which we need to observe carefully. So now which pigments are involved? From pigment part, action and absorption spectrum thing question can come, but as I said, light reaction mainly we need to understand where what is placed. But yeah, you should have an idea of these pigments first. So let's get started. So photosynthetic pigments, what exactly they are? They are basically beta the proteins, right? They are also the protein complexes which can trap the light energy. And when they trap the light energy, the electrons will get excited. So it is ultimately the electrons movement that you study in the light reaction, in which you study the Z scheme. Are you getting my point? There you study that. Are you getting my point? Are you getting my point? So now come back here. Let's have a quick revision. Let's pay attention, guys. So you people can see when you are talking about the photosynthetic pigments, है ना? So special molecules that absorb, right? Special molecules that absorb, transmit and reflect different colors of light in the visible spectrum. Is that clear? In the visible spectrum. So it is the power, you know that from that sunlight, right? 50% of the light, what will happen? Out of 100% incident light, right? It is only the 50% of the light that will be trapped. Out of that, 2 to 10% is photosynthetically active radiation. So they are in this particular range, 400 to 700 nanometer. Am I clear here? Yes, everybody, show some energy here in the chat section what you people are doing. And if you are new to the channel, you know what you have to do. Quickly hit the subscribe button. Quickly hit the subscribe button then. And if you are new, just quickly hit the like button as well. How can you forget about that? Quick. So basically PAR, photosynthetically active radiation. Here you have 400 to 700 nanometer. So you are talking about the Vibgyor here, right? Vibgyor here. That's what you need to talk about. So you have violet, the indigo, blue, there will be green, there will be yellow, then orange and red. That's what we have. Am I clear? That's what we have. Vibgyor. So let's talk about the pigments. So chlorophyll, carotenoid, phycobilins are there. Let's understand about them. So chlorophyll's major pigment. This is my direct MCQ. Please write down this line. Everybody, please write down this line that light is required for the synthesis of the chlorophyll. If there is no light, the chlorophyll will not get synthesized. From the experiment parts, we focus on that. So that's how you have to remember. So chlorophyll, green colored pigment, light is required for their synthesis. So they are soluble in the organic solvent, but this is the main point, that it is the light which is required for the synthesis. So if there is a question that if a plant is not getting the light, the chlorophyll will synthesize. Yes, Shravani, Shoaib, chlorophyll will synthesize? No. Chlorophyll will not synthesize. So chlorophyll is of different types. We have already completed that in the previous marathon. A, B, even we have chlorophyll C, chlorophyll D, E is also there. Just if you remember, you will find A in all. So it is the major photosynthetic pigment. Do you remember that? So it is the major photosynthetic pigment. It is also known as universal pigment. It is also known as universal pigment. Now, the next is chlorophyll B. So A and B, you will find that in green algae, beta. You will find that in green algae or in higher plants as well, right? Or in higher plants as well. A and C, if you remember, brown algae. That's again a repeated question. A and D, that will be in the red algae, that is your rhodophyceae. So this is also the detail. So A is the major photosynthetic pigment, right? A is the major photosynthetic pigment, the universal pigment, right? So that's what you have to keep in your mind. So this is what you have to remember, guys. So just keep it in your mind. So this is the universal pigment in all oxygen liberating photosynthetic organisms. Is that clear? Is that clear? So chlorophyll A is having blue-green color in chromatogram. Another important question. Jaise beta, we have a technique for here. We go for paper chromatography. right? I will not explain that in detail, but just say, let's suppose if you have a green leaf, it doesn't mean that it is just having the green color. So basically in the chromatography, you separate, right, the different components of this, right? Right, you will separate the different components. So when you separate that, you need a mobile solvent, right? You need a mobile solvent, you need a place where, just say, you might have seen that in the lab. So in a very simple way, I'm trying to explain you that, like, let's suppose there is a container, a paper will be there, you will take the leaf, you are going to mash them, you will make that color, the this kind of liquid, or I would say the kind of slurry. So you will put that on a paper. Now there is a solvent as well that will move, right? Slowly it will move. It will separate that this green colored, slurry. Yeah, I would say that the green colored kind of mixture into different of its component, right? So this is the chromatography which help us to separate them. Are you getting my point? So on that chromatogram, which pigment is having what color? That is again an important question, right? So in my regular lectures, I also explain what chromatography is, what paper chromatography in the sessions itself. But now I told you time constraint is there. Just stick to the topic so that we can get best out of that. Focus on one thing, that it is the paper chromatography which we are using for the separation here. Are you getting my point? That it is a paper chromatography that we are using for the separation here. So that's what you are supposed to remember. And here on the paper, we have what? On the paper, we have what? We have on the chromatogram, on that paper, we will see, right, the different colors here. Right. So here for the chlorophyll A, it's going to appear like a bluish green. So this is the formula C55H72 is there, O5N4Mg. So now, this is the structure. So for chlorophyll, I'll write here, you already know that for its synthesis, what do we need? For the synthesis here, right? For the synthesis here, you know that light is required, yes. Even beta, your glycine, your succinyl coenzyme A, all these names are not new for you. They are required, right? And for biosynthesis, please write down everybody. For biosynthesis, iron is required. Fe, it is pronounced as iron. Iron is required, right? And central metal ion. But central metal ion is magnesium. Okay, it's magnesium. So for synthesis, you need iron, that is iron also. We pronounce it as iron also, but actually it is iron. But for the central metal ion is magnesium. Now when you look at the chlorophyll structure, so in the case of chlorophyll, first of all, you will see this part. Like, it is believed that its structure look like a tadpole. Tadpole is your frog's larva. Tadpole is your frog's larva. Right? There's a head will be there, your tail will be there, something like that. Yeah, you can even make it head is there or tail is there, like that. Now, when you look at this particular head, what exactly it is? It is tetrapyrrole. Tetra means 4 ring. Or this is basically porphyrin head. Sorry, porphyrin head. Porphyrin head. For head, I'm writing red. Matlab, I need a T. Porphyrin head. And this one is what? This one is my phytol tail. So porphyrin head and phytol tail is there. So central metal ion is magnesium. This is important PYQ. So this is your first tetrapyrrole. Out of these tetrapyrrole ring, this is your first one. This is the second one, third one, and fourth one. Right? So this is your phytol tail, carbon, hydrogen and all. So structure is not very important to remember. Central metal ion is important. For biosynthesis, ion is required, important. Glycine succinyl coenzyme A is required for synthesis, important. Light is important. Structure, it's okay, no worries. Now when you look at this, in the case of chlorophyll A, here in the second pyrrole ring at this position, you will have CH3 group, that is your methyl group. And in the chlorophyll B, you will have CHO group, that is your aldehyde group. Important. Another important topic. Quick. Another important topic, guys. Quick. Yes, quick. Done and dusted. That is the another important thing. So now don't get confused ki madam that time jaise you can see that first, second, third or this one is the fourth. So second mein you will have in chlorophyll A methyl group, but in chlorophyll B at same position you will have CHO. So it is having like 2 extra hydrogens and it is having 2 less hydrogen but 1 more oxygen. Okay. Okay, so the structures are literally very important. You are not allowed to miss it. You are not allowed to skip it. So second pyrrole ring mein you will see, and which color do they have on chromatogram is another important point. So it is yellowish green in chromatogram. So it is basically the accessory pigment. Now this will help us to understand the photosystems. So please pay attention guys. Please pay attention. Okay. Now come back to this. I already told you looks like a tadpole. Yeah, so already told you, looks like a tadpole, porphyrin head, it is water loving, tetrapyrrole structure with magnesium at the center. So central metal ion is magnesium, but for the biosynthesis you need iron. Okay, so phytol tail is there beta, and this is hydrophobic embedded in that thylakoid membrane. So precursor, you know that protochlorophyll is there. So this is the reaction, succinyl coenzyme A plus glycine, iron and light is required. You will have protochlorophyll and then you will have chlorophyll. Any doubts? Any doubts? Any doubts? Sure? Are you sure? So that's what we have to pick up. That's what we have to take care. Next is the carotenoid, another important topic. So they are also lipid soluble. It is your phycobilins that are going to be water soluble. So this is also important. So in green and non-green, both plant parts will have that. So they absorb light energy. They pass it to the chlorophyll A. Why and how? We'll talk in the PH2. In photosystem. So they are the accessory pigments. Just chlorophyll A is the main pigment. Rest all the pigments that we are going to discuss, they are the accessory pigments, shield pigments, right? And their job is to, absorb the wavelength of different different wavelengths of light and finally to pass that energy to chlorophyll A. So chlorophyll A also absorb the light directly, but other pigments also pass the absorb light to it so that it can provide it the more energy so that it can excite its electron. That's how the complete light reaction occur. So that's what we are going to talk about. So protect chlorophyll from photooxidation. So it will protect it from the oxidation by light, right, by squelching this nascent oxygen and by converting it into the harmless oxygen. Right, done. So that is why they are the shield pigment, या फिर even we can call them as the accessory pigments, the yellow to yellow orange pigments. That is how they look on the chromatogram. So they are in the chromoplast also, chloroplast also. So they are not present in eubacteria. This is the most stable pigment where the light is not required for the biosynthesis. So I believe this question is important. So in general, beta, questions will come from whatsoever topics I have suggested. Even yesterday also I posted a strategy, right? How many of you have seen this? Yesterday I posted a strategy. Have you seen that? That particular strategy? Huh? In which I spoke about the last 15 days plan, the most important plan, I would say. The most important plan, because I told you there that for getting 600 chapters, you need the full command over 600 marks, you need the full command over 55 chapters. I told you that which biology chapters you need to remember, you need to study, and even I told you there, right, which physics and chemistry chapters you need to master. Right? So if you have seen it, very good. If not, just go and check. Okay, after the class basically. So this is one thing that you have to understand. So general topics are same beta. Whatsoever we are saying, this will come, that will come, this will come, that will come, that is all same. But beta, point is na, points like this, these are the game changer. They boost your score. Jaise chlorophyll ke liye, you need the light. If there is no light, it will not get synthesized. But for the carotenoids, there is no requirement for the light. Even if light is not available, still their synthesis will occur. still their synthesis will occur. Is that clear? Chaliye. So chemically they are also the terpenes. So they have two types. One is carotenes, another is xanthophylls. Right? One is carotenes, another is xanthophylls. So that is what you are supposed to remember. Right? Carotenes and xanthophylls are there. So this is yellow orange colored, important. So जैसे carrot है ना, carrot, your beetroot, they have beta carotene. This is the example. So beta carotene is very important, beta. It is also the precursor of vitamin A. It means it makes the vitamin A. Am I clear? It's the precursor of vitamin A. Is that clear? Is that clear? So it is the precursor of vitamin A. Got that? Very good. Excellent. Excellent. Beetroot mein, carrot mein you have that. Xanthophyll, they give yellow colored. Carotenes, yellow, orange. This one yellow, right? So xanthophyll is having different types. This you remember? Brown algae. This is again a repeated question. Brown algae. Brown algae is brown due to the pigment fucoxanthin, right? So fucoxanthin is also a type of xanthophylls, yellow color. So it is also a type of xanthophyll. So during the autumn foliage, during the autumn season, we see beautiful yellow colors, right? So ye dekho, lutein, again a type of xanthophyll. Okay, okay. So these are the facts that you have to remember. So next is, this one I have already discussed, phycobilins. So another fact here that we have is this, hot water soluble, right? जैसे phycocyanin, cyanin word is associated with blue, phycoerythrin. जैसे your red algae is red due to this. So I believe this part, this portion is clear to you. So that's it. So phycobilins are hot water soluble. Yeah, water soluble they are. Your lipids, your carotenoids are also the lipid soluble, right? Your chlorophylls, organic solvent soluble. So light is not required for the carotenoids. Light but is required for the chlorophyll synthesis. Am I clear? Pakki baat? Sure? Sure? Okay, done. So now structure sub done, hai na? So by chromatography, this is what you have. So this is by paper chromatography. Guys, take the screenshot right away. Take the screenshot right away, the most important one. Quick. Just take the screenshot right away. Guys, quick. Quick. The most important topic. One question we can expect from this part, even in match the column format. How can we expect this? Even in the match the column format. Guys, quick, speed up. Do it. Okay. Chaliye, let's go. So these are the various regions, various lights. Am I clear? Chaliye. So now the next thing is, ये देखो, see this. मतलब जैसे we say ना 400 to 700 nanometer या 390 to 760 nanometer, मतलब this is the range, this is in the angstroms. Now red light above, okay, if it goes above 700, right, या फिर somewhere around above 680 or something, far red. If it is below 390 nanometer, UV rays, you have studied that in physics and I assume that you know that. Beyond red, मतलब beyond this 760 आ जाएगा, infrared. So this is the range. So in this part only photosynthesis will be there. So now that's the topic. How many of you, these graphs I'm going to resolve for you. How many of you find it difficult? Quick. After these graphs na beta, I'll tell you what PS1, PS2 is. Then directly I will teach you what Z scheme is. Dark reaction is not at all a cakewalk. I'll just make a table and I'll elaborate that. So how many of you find it difficult? Accordingly, I'll see, should I explain it or should I skip it? Batao bhai, jaldi, quick. Yes. So those who want me to explain it in depth, I want to see some fire in the chat section. And if you people are new to the channel, you know what you have to do. Hit the subscribe button, guys. Hit the subscribe button, guys. Quick. If you are new to our channel, just hit the subscribe button. Don't forget to do so. Quick. Waiting, waiting. Pakka. Who wants me to explain this? So beta, basically we are growing this channel like a little baby, hai na? So already I have given many years on some another channel as well, which we have grown, right? Now it is the time for this particular channel which we have to grow. And yeah, because of you people, because of our lovely subscriber, because of our lovely students, yeah, we can even see an increase. So I am not expecting much. I am expecting it, okay, we will grow slow, but I really want that in next 5 days, at least, yeah, we have to cross this 10,000 ka number. My wish is to reach to 15,000, right? And then till NEET, you people will definitely raise it to the 50,000, right? 15 to 50. Dekh rahe ho? Expectation, positivity, ki we will do it. So that is what I'm expecting from you guys. Okay, right? That is what I'm expecting from you people. So jaise we teachers, yeah, if I'll talk about myself, that I don't I don't believe to stay in my comfort zone. I keep challenging that always, because I believe when you leave your comfort zone, then only you get the growth. Otherwise, there is no growth, right? So you people are still students. You are young. You should have more enthusiasm than me. Right? You should be ready always. Okay, ma'am, we will get out of our comfort zone. So if you people are scared, "Arey yaar, ma'am, 3 hours are done." Just 3 hours. Imagine, just 3 hours. Right? We have to read. We have to read up to like I am to planning till 11 p.m. so that we can finish, we can get everything out of that. And trust me, if you will watch these 4 marathons of mine, right, nobody can stop you to score 320 marks, because I'm covering everything. So yes, help us to grow, right? That is your duty. I want you people to grow us, help us to grow, so that we can provide you the quality content. And imagine if we are doing that much for our NEET 2026 aspirants, right, where we just have few days, what are we going to do for NEET 2027 aspirants? Just imagine once. Okay, so keep supporting, keep growing. Let's come back, let's discuss that. There are 3 graphs which we have. There are 2 things that I have to, or we have to understand here. The first one is absorption spectrum. The second one is action spectrum. The first one is absorption spectrum. The second one is action spectrum that I have. Guys, I cannot see the charts. Are you there? Bhaiya, show some presence. Hello. So, absorption spectrum is there, action spectrum is there. Word itself is saying absorption. Word itself is saying absorption. So it tells us that which pigment will absorb which wavelength of light. Which wavelength of light is absorbed maximally, yeah, which wavelength of light is absorbed less. So all that things we have here. That's what we need to understand. So this particular graph, firstly, discuss it in parts and then we will go ahead. So it is your absorption spectrum. It is your what? Absorption spectrum. This one is telling me about the action spectrum. It is showing me both. Now, let's revise. Let's revise. Done? Done? So, absorption spectrum mein, can you see that? Ye dekho. Chlorophyll A. है ना, blue green, green, yellow. Now in this region, can you see the peaks? I'm teaching you from the scratch. I am teaching you from the scratch, right? I'm teaching you from the scratch. Can you see that? Done? Done? Quick. So you people can see that here, you can see here, you can see the peaks. Here also you can absorb some, observe some peaks. Here there are no peaks. Here there are no peaks. This is what we are, looking at. Yes or no? This is what we are looking at. Yes or no? Yes or no? Yes, everybody. है ना? Quick. So one is absorption. Yes? Exactly. So absorption spectrum is there, action spectrum is there, right? Absorption spectrum is there, action spectrum is there. So that is what you have to observe. Okay, that is what you have to observe. So you can see peaks are in this region. Why? And now this is the action spectrum. It tells us in which light the rate of photosynthesis is maximum. Action spectrum tells us that in which light the rate of, right, the rate of, I would say, the photosynthesis is maximum. This is the action spectrum. Here there is the comparison. So if you will see, just 400, 500, 600 nanometer, 700 nanometer. So don't you think that this part is showing me the blue region, blue part of, जैसे we have Vib-Gyor, violet, indigo, blue, green, yellow, orange and red. And then here what do you have? Here you have the red. Right? Here you have the red. Are you getting my point? Are you getting my point? So this is what you need to focus, and that's how you have to emphasize here. So now let me elaborate that. Again, just have a look. Violet region, blue region, and then you have the red region. So these are the, I would say, the most important topic or the gist there which you need to understand. So blue and red light. Blue and red light. They are most effective for photosynthesis. They are most effective for photosynthesis. Yes or no? They are most effective for photosynthesis. This is the first thing that you have to remember. Is that clear? Then, green light, least effective for photosynthesis. Least effective for photosynthesis, right? Least effective for photosynthesis. Blue and red light is most effective. Out of blue and red, it is the red which is even better than blue. That is what, like, both are there, but still red is more effective. Is that clear? Green is least effective. Why green is? This is again the most important portion of photosynthesis part, from where the direct MCQ is expected. Is that clear? So this is again the most important part, from where the direct MCQ is affected, is there, right? Is there. Is that clear? So green is the least effective. Why? Because plants, can you see that? They are not absorbing the green color. You find them chlorophyll, green in color. Chlorophyll is required for the photosynthesis. Leaves, when you look at them, they are green in color. So what is the point there? What is the thing that you have to focus there, right? What is the thing that you have to focus there? That green color. Because why? Why do we see that something is green? Yeah, something is red. Yeah, something is blue. Because they are reflecting that color. So if leaves are green in color, it means they are reflecting green color and they are absorbing rest of that. So that's why green color is the least effective for the photosynthesis. That's why those, small, I would say, the herbs, yeah, even for the shrubs, they grow under the large trees. Yeah, if there is a tree of large canopy under that, if there are small trees, they don't get, right, they don't show the high rate of photosynthesis. Why? Because they are under that thing, na? So this greenish color they are getting, that light they are getting. So that's the problem. Are you getting my point? That's the problem. So that's what you have to remember there. Are you getting my point? So red, blue and red is the most effective one. Green is the least effective one. This data is very important, and absorption spectrum is telling us this thing, कि leaves appear green because chlorophyll do not absorb green color, they reflect it, transmit it. Blue and red region most effective. Blue light carries more energy. जैसे red light का wavelength is more, but blue light carries more energy. Red light carries less energy. But which one is most efficient? Which one is most efficient? So which one is most efficient? You people can see that Also, one second beta. Again it is coming down. I don't know what is the issue here. So here you people can see red is having less energy, but it is most efficient in the photosynthesis. Blue light carries more energy, right? So green light is least effective. So that these are the points which we have to remember. So this is your action spectrum. So if you look at the action spectrum, beta, if you look at the action spectrum, then what do you see? If you look at the action spectrum, what do you see? You people will see this particular thing, that when I look at this region, right? Dekho, look at this. Try to decode it. So in red or the blue light, maximum peaks are there. Red and the blue light, maximum peaks are there. Is that clear? Is that clear? So you people can see that. Now, if I have to depict it, only this portion, can I not say that chlorophyll B, firstly, chlorophyll B absorb more blue light, B for chlorophyll B, B for blue, light, then red, number 1. Chlorophyll A absorb more blue light, then red. Number 3, chlorophyll B absorb more blue light than red light. Chlorophyll A absorb more red light than blue light. Beta, this part, this segment is literally very important. Then carotenoids showing no peak in red region. Now tell me if there is any doubt. If there is any doubt, do let me know. Quick. Matlab, whatsoever these points are, they are directly from the graph which is given. Done. Quick. Yes, beta. Done. So whatsoever I have mentioned, is it correct or not? Or is there any mistake? Figure out. So dekho, chlorophyll B is absorbing more blue than red, true that. Chlorophyll A is absorbing more blue than red, true that. But if I compare, chlorophyll B absorb more blue than chlorophyll A, and chlorophyll A absorb more red light than more red light than, my bad, chlorophyll B. Dekha, I asked you to check that. And then carotenoid showing no peak in the red region. So that is what you have to understand, right? Both second and third point say the same thing. Now see, chlorophyll B absorb. Nahi beta, chlorophyll B is absorbing more blue light than acha. Okay, got that. Then chlorophyll A. Very good, Aditya. So then chlorophyll A. Now clear? Now I have rectified them. Okay, done? Is it right now? है ना? Exactly, very good. So, which one is the most effective? Red and blue. Out of red and blue, if I have to focus, red. Okay, both are very important. So that's what you have to remember. So obviously, accordingly, the action spectrum is going to be, so curve, action spectrum is the curve showing rate of photosynthesis at different wavelength. So which one will show the maximum? Red and blue. Okay, so here you can see, ye dekho, look at the gap. Blue light is absorbed more comparatively, look at the rate. Red light is absorbed less comparatively, look at the rate, look at the peaks there. That's how you have to retain. This is it. So this is the another important topic. Now I believe after understanding this part is clear to you. Yes, I believe now this part is clear to you. Yes, beta. Sure? Sure? Okay, absolutely right. Chali. So next is quantum yield. Quantum requirement. Yield means what you have made, what you need. Just say number of light quanta which are required for production of one oxygen molecule, 8. So if 8 oxygen molecules you need for the formation of, for the formation of one oxygen molecule, if you need 8 quanta, so obviously number of oxygen molecule evolved per quanta will be 1/8. Simple mathematics. Simple mathematics. Am I clear? Now this portion is not given in the this part. This portion is not given in the NCERT, but this is very important to understand. Jaise many scientists, jaise we will talk about the immersion effect here, red drop effect. So some scientists, they absorbed one thing, right? Some scientists, they did what? They absorbed something, that when they are illuminating the chloroplast, when they are providing the chloroplast a light, so they were absorbing key when they were showing the maximum rate, less rate, like that they were doing. Okay? So when chloroplast, they are illuminated with the far red light, which is beyond 680 nanometer, usually around 700 nanometer, the quantum yield drops sharply. When it is around 700 nanometer, quantum yield was coming less. This is what they observed. They called it as the red drop. When they have provided the far red light, something which is above 680 nanometer, something which is above 680 nanometer, usually around 700 nanometer, this is what they observed, that there is a drop. So they called it as red drop, right? They called it as what? They called it as red drop. Now there is immersion enhancement effect also. So in 1957, Emerson observed that if red light around 680 nanometer and even far red light, here what they did, they just provided light above 680 nanometer, around 700 nanometer. Just that. Drop was there in the photosynthetic yield. But when they provided both, 680 nanometer bhi, even up to 700 nanometer, 700 nanometer bhi, then they were like, when both were given, then rate of photosynthesis was greater. So this phenomena is immersion enhancement effect, that when you are providing 680 or 700 nanometer both, and this led to the discovery of the two photosystems, right? This led to the discovery of the two photosystems. So now we have started the most expected topic for today's lecture. Ready? Now we have started the most expected topic. Ready? Ready, ready, ready? Pakka? Sure? Okay, chali. Now, we have something known as photosystems. What are they? Beta, nothing. They are the group of pigments itself having two parts. One is reaction center. One is reaction center. And the another one is what? One is reaction center, and the another one is light harvesting, LHC, light harvesting complex, which is in short LHC, or you can also call it as antenna complex, which is also called as antenna complex. In the reaction center, you will always get right? You will always get what? Chlorophyll A. right? You will always get chlorophyll A. So you have light harvesting complex, which is also the antenna complex. Here you have other pigments other than chlorophyll A. Pigments other than chlorophyll A. That is what you have. Pigments other than chlorophyll A. That is what you have. Like other than chlorophyll A, right? Other than chlorophyll A. Is that clear? Is that clear? Right, beta? Whatsoever pigments are there, chlorophyll B, carotenoids, all your xanthophyll, carotenes, yeah, even phycobilins, they all will be present here. So these are the two components of the photosystems. So you keep it like this, ki this is the reaction center, the main center, where you just have chlorophyll A, and then there are other pigment molecules also. Their number can be 100 to 120, like it is like this. So they are in the nearby, they are light harvesting complex, yeah, antenna complex. It is not like that reaction center, the chlorophyll A cannot absorb the sunlight. They can also absorb. But here see, we have a chlorophyll A, we know that in this particular wavelength of light will be absorbed by it. Now in the light harvesting complex, because this is having the accessory pigment, this is having the shield pigment, this is having the other pigments, so they can even absorb different wavelengths of light. Just say your carotenoids, right? You have seen them in the blue region, right? But you will also see different types of carotenoids are there. Some will absorb, some can even absorb yellow light, some can even reflect that. Like different ways are there, right? So they will absorb the light of different wavelengths. So what will happen when they will also absorb the light, they will also pass it to each other, and finally ultimately they all will pass it to the reaction center. It will not go anywhere away. It will only pass to the reaction center, and from the reaction center, right, chlorophyll A will take it, and then the electron will excite. That is how it works. Now, and plus they will make sure that the photooxidation of chlorophyll A should not occur. So we have two photosystems. We have photosystem 1 and we have photosystem 2. But beta, their number is as per the order of their discovery. Their number is as per the order of their THEIR wala their discovery. As per the order of their discovery they have. As per the order of their discovery. Jaise PS1, we found it first. PS2, we found it later. PS1, we found it first. PS2, we found it later. Are you getting my point? So it's not like that ki it is as per their function. If I talk about their function, so we will go like this. PS2 to PS1. We will go like this. Electron moves from PS2 to PS1. So why? So this PS2, it is also referred to as P680. PS1 is also referred to as P700. Why is it so? Because it will show the absorption peak at 680 nanometer. It will show the absorption peak at 700 nanometer. Is that clear? This one will show the absorption peak at 680 nanometer. This one will show the absorption peak at 700 nanometer. So that's what you have to remember. Am I clear? So this is what PS1, PS2 is. Now, their locations are very important. So you will find your PS2 only in the appressed part, the one which is stacked, which is not exposed. PS1 in the non-appressed part plus in the stroma thylakoid. So in stroma thylakoid, again direct PYQs, in stroma thylakoid, you people will see, what will you see? That even PS2 and FNR, yes, ferredoxin, NADP reductase, right? Ferredoxin, NADP reductase. This is absent. That's important information, guys. So grana thylakoid mein you have both, but stroma thylakoid mein you have only one. Most important information. So tell me quickly. Done? Okay. Sure. So this information is very important. Okay. So this is how your PS1, PS2 will help. Located at this and that part, reaction center kya hai? So this PS1 is of reducing nature, oxidizing nature, participate in both cyclic and non-cyclic photophosphorylation, but this one will participate only in non-cyclic photophosphorylation part. Just have a look. Just have a look. Beta stroma thylakoid, stroma lamella, fret lamella, your fret, this intergranule thylakoid, they are all same. They are all same. So quick, everybody have a look. Quick. Yes. Done. So this is important question can come from this part. Now I'll conclude it with the light reaction, and then we'll pick up the important topics from the dark reaction, and then we will move to the respiration part. Okay. So at 1:30 we started it now. 1:30, 2:30, 2:03. This one will like we will go till 3:45 to finish photosynthesis because it is lengthy. Respiration will not take much time because there also I will teach you the selected topics, selected subjects, selected topics, selected steps with the help of tricks, right? So I think you guys have seen that even in the shorts also, right? Yesterday I posted a short regarding the Krebs cycle trick. Okay, chale. So come back here. Let's discuss it. Dekho. When it comes to the light reaction, you already know about its products. You already know where will it occur. Now, here you will discuss it in this way. Just say one is non-cyclic photophosphorylation. One is non-cyclic photophosphorylation. Another one is cyclic photophosphorylation. Now, the very first point I would like to specify here is, chalo, firstly I'll tell you what is cyclic and non-cyclic. In any non-cyclic process, say you are starting from A, you'll go to B, you'll go to C, you'll go to D, you'll going like this. Done. In the cyclic process, you're starting from A, you're going to B, you're going to C, C is coming back to A. This is the meaning of a cyclic process. So whenever there is any cyclic process, so from where you start this process, the cyclic process, you need the replenishment of that. You need that particular thing. You need that particular compound. It is mandatory. Okay, it is mandatory. Now, the next word that we have to understand here is photo, that is light, phosphorylation, mane addition of phosphate. So it means, right, that you will make ADP plus PI will give you ATP in the presence of light. Means you need light so that you can add this inorganic phosphate to this ADP and so that you can make ATP. So three types of phosphorylation. Three type of phosphorylation is the part of our syllabus. Photophosphorylation, where you are using the light energy to make the ATP. So by using light energy, you are adding the inorganic phosphate to that ADP so that you can form the ATP. Right. Second category is of substrate level phosphorylation, and third category is of oxidative phosphorylation, that also we will discuss later. Is that clear? Is that clear? So that's what you all need to revise, you all need to understand. So this non-cyclic photophosphorylation is your Z scheme, and here you need both PS2 and PS1. Here only PS1. Doubt? Doubt? Hanji, doubt? Doubt? Sure? Only PS1. Is that clear? Is that clear? Pakka? Sure? So, dekho. This is your thylakoid. Matlab in thylakoid membrane this process will occur na. Thylakoid. This is your thylakoid lumen. This is your thylakoid stroma. So beta, actually in this part na, in this membrane portion, you have different pigment molecules. ठीक है. Here there is a list, here there is a sequence, and in which there is the electron flow. Let me discuss that. जैसे let's suppose you will see PS2 is there. You will also see PS1 will be there in obviously in non-oppressed part. You will see meanwhile there are different electron or hydrogen carriers are also present. So there is a kind of, you know, arrangement of all that things. Right? In simple way, firstly, I'm telling you this. In a very simple way. Just allow me 1 minute, please. Yes, everyone. So am I audible? I believe I'm perfectly audible. So that is how their arrangement is. So that is how we will see that how the electrons will flow and things will go on. So if this thylakoid membrane is clear to you, right? If this thylakoid membrane is clear to you, the structure. So can I not say that, guys, can I not say that this side will have, wait, this side will have lumen and surrounding to it, there is the stroma. Yes or no? So we will firstly start this process from PS2, right? We will talk about the pheophytins, which are somewhere associated here only, pheophytin. You will talk about the plastoquinons, plastohydroquinone, cytochrome B6, F complex. We'll talk about the plastocyanins as well. We will talk about the plasto PS1 as well. We'll talk about the FD, FNR as well. And there will be this, like this. Now, I'm just simply telling you an arrangement. Okay? Matlab, this is what you have to remember. You can even, try to revise it or try to remember it with the class itself. PS2, pheophytin, plastoquinone, plastohydroquinone, cytochrome B6, F complex, plastocyanin, PS1. Let's break it. Let's try to remember it. And then I will move to the next topic. Is that clear? Then I will move to the next topic. Is that clear? Is that clear? Quickly tell me. Yes, please. Done? So that is the sequence that we have to remember. I am writing it here. PS2, Theophytin. You firstly try to remember that, then I will elaborate it. Then you have plastoquinone, right? Then you have, I'll tell you why quinone, plastoquinone, plastohydroquinone, which is also known as plastoquinol. Then you have cytochrome. Firstly, try to remember this much. B6F complex, and then PS1, unknown electron acceptor A, B, chalo, that I will discuss later. So ferrodoxin, NADP reductase as well. Done? Done. Is that clear? So this is the sequence that you have to remember, right? So PS2, pheophytin, plastoquinone, then plastohydroquinone, then plastoquinol, then cytochrome B6F complex, photosystem, one thing is missed here. Yeah, then plastocyanin, then PS1, then ferrodoxin and ferrodoxin NADP reductase. Try to remember it. If this flow is clear to you, then Z scheme is not going to be any problem. So I'm repeating it again. We have photosystem 2, we have pheophytin, we have plastoquinone, we have plastohydroquinone, which is also known as plastoquinol. We have cytochrome B6F complex, then we have plastocyanin, PS1, that is photosystem 1, then unknown electron acceptors are also there. Finally, ferrodoxin, then ferrodoxin NADP reductase. So those who are done with it, do let me know in the chat section that ma'am, it is done and dusted, and quickly hit the like button. And if you're new to our channel, subscribe to this channel. Quick. Then subscribe to this channel. Quick. Yes, please. Yes, please. So again, I'm repeating myself. We have photosystem 2, we have pheophytin, we have plastoquinone, plastohydroquinone, which is also known as plastoquinol. Then you have cytochrome B6F complex, plastocyanin. Photosystem 1 is also there. Then ferrodoxin, NADP reductase, and ultimately NADP. Pakka? Sure? Want to see some fire emojis here in the chat section, guys? Speed up. Want to see some? In parts we are going to discuss that. We are not going to discuss it in one go. In parts I'm going to break it. Firstly, what we read the meaning of. Let's summarize. Firstly, we read the meaning of, like, what do we have in the light reaction. Non-cyclic photophosphorylation, we are also calling it as Z scheme. Both PS1 and PS2 and PS1 are present here, right? So A, B, C, D, E, this is what we have here. That's what I told you, number one. Then I told you cyclic photophosphorylation is there, only PS1 is there. Then we discussed that there are the protein complexes, electron carriers or the hydrogen carriers which are present here in the thylakoid membrane, right? I told you PS1 will be in the non-appressed part. That's what we have to remember. Then in the next part, I told you their location. This is the lumen side. Even if you will carefully observe this particular diagram, it is also telling you, just say plastocyanin is towards lumen side, ferrodoxin is towards trauma side, FNR is towards trauma side. So this is not something that I loved, ki, "Arey yaar, I will make it like this." No, it is about their location as well, that we have discussed. So PS2 hai, pheophytin hai, plastoquinone hai, plastohydroquinone hai, cytochrome B6F complex, plastocyanin, PS1, ferrodoxin. In between, unknown electron acceptors are also there. वैसे in detail in higher classes, so we also study the quinone, how that plastoquinone is present, how are the like different ways are there, but right now time limitation hai, topic limitation hai, so it is like that, and that's how they are present. That's how they are present. So their position is also very important, guys. Their position is also very important. And ultimately, what's going to happen here? Electron flow. Why that electron flow is important? Because we need to maintain a gradient. We need to maintain a proton gradient that will help us to make the ATP that we understand in chemiosmotic hypothesis. I told you initially, light reaction will give me ATP, will give me NADPH2, will give me oxygen. So now we have to see where are we going to get what. So this is thylakoid membrane, this side is lumen, this side is stroma. Our agenda throughout is what? We have to maintain a proton gradient, right? So now I'm telling you agenda. Agenda is we need proton gradient. We need more H positive. We need more H positive inside lumen than in stroma. That's what we need. So that's what we have to keep in our mind. That's what we have to keep in our mind. Is that clear? Is that clear? Is that clear? Done? So we need more H positive inside lumen and we need lesser than that in the stroma. Is that clear? Right, look at the position, look at the position, look at the electron flow of also. That's why I told you as per the order of discovery we say 1 and 2, but electron flow is from PS2 to PS1. Electron flow is from PS2 to PS1. Finally, we will talk about the CF0, CF1 that will help in ATP synthesis. But meanwhile, this is the portion that we have to target. So yes, students, you should know PC, plastocyanin, copper. It is definitely having the copper towards lumen side, ferrodoxin towards stroma side, FNR towards stroma side. That has to be clear in my mind. That has to be clear in our mind. This is something very, very, very important. This is something very, very, very important. Is that clear? Is that clear? Right? Dekho, this is how these complexes and all are arranged. So yeah, there is one electron transport chain that you will read here also. So now, this is one point, one diagram. Firstly, tell me this part clear. Firstly, tell me up to this part clear. Then I will tell you why do we call it as the Z scheme. Then I will tell you that why do we call it as the Z scheme. Quick. Quick. Yes, quick. हाँ जी. पक्की बात? Sure? Done? चलिए, let's move further. Okay. Now, we are calling it as a Z scheme. So, this Z is concerned with what? Like, yeah, you can keep one thing in your mind. Something is in zigzag manner. Why zigzag manner? Because here the things are as per their redox potential, which you also call as oxidation reduction potential. So, it is as per your redox potential, which you are also calling as oxidation reduction potential. Now, let me explain that. Just say, let's suppose this is a particular entity A, this is a particular entity B, right? This A is having high redox potential. Try to understand that. This A is having high redox potential. This B in comparison to A is having low redox potential. So any entity with more, this is the general phenomena that we have. Any entity, any particular entity with more redox potential, with high redox potential, any entity with more with high redox potential is capable enough to accept the electron. So out of B and A, the electron will move to the A. Why? Because its redox potential is high. Now let's suppose you have C. C's redox potential is higher than A. So if it is higher than A, this electron will move to this. So that's the basic funda. That's the basic funda that is going on. Each and everything that I'm explaining right now, beta, each and everything that I'm explaining right now will help you to understand this topic in depth. Even if you are watching this for the first time, you can understand that. I am not at all compromising the content, right? Even if I have to take the session for 12 hours, 13 hours, 14 hours, it doesn't matter to me. All I want is I will not compromise the content. 4 marathons, 320 marks for sure. Done? Done, done, done. Sure? Are you sure? So redox potential, this is what we are discussing. So that's how the electrons will move. So this is basically, in the case of redox potential, this is actually down the gradient, right? Down the gradient. Yeah, you can say that this is actually the downhill movement. Yeah, down the gradient. Yeah, you can say that this is actually the along the gradient wala thing. Jaise in osmosis, yeah, in diffusion, jaise in osmosis, what you people see, that water will move from its higher concentration to lower, right, through a semi-permeable membrane. So this is the down, yeah, along the concentration gradient, because here you will see that water will move from high to low. Don't confuse, don't focus on high to low wala word, don't just stick to the high to low thing, right? That is not what I am trying to tell you. Obviously, if water is higher at some place, it will go to that lower thing. Solvent is more there, solvent will go there, but that's not what I'm trying to tell you. And see, now in the case of redox potential, that's how the things are, right? Now in the case of redox potential, what is the scenario? What is the thing in the case of redox potential? The thing is very simple, beta, right? Thing is very simple. Obviously, if there is an entity, just say, let's suppose if this is one entity that I have, this is another entity that I have, this entity is having more redox potential, this is less. Who deserves? Who deserves the electron? Who deserves the electron? This is having more redox potential. This one is having less redox potential. This remote is having less. This bell is having more. So which one? To whom you should think that electron should go? Quick. To whom you think that electron should go? More, less? Yes, please. Hanji, where? Here? Yeah, here. High redox potential, low. Hanji, obviously, beta. Obviously, this one, right? Because its redox potential is high. So it is having more tendency to accept the electrons. Are you getting my point? So that is what you have to understand, right? So electrons will pass to the entity having more redox potential. Just in my case, this is the entity having more redox potential. Are you getting my point? So obviously, in the case of electron movement, electron should pass from any entity having low redox potential to another entity having high redox potential. So that will be there down the gradient, yeah, downhill movement. It is going to be like this, right? But if electron pass from any entity having more redox potential to less redox potential, then you will call it as uphill transport. Uphill transport. Is that clear? Yeah, uphill movement. Now, so in this particular Z scheme, all the things are arranged in this way. Matlab the things, the way these protein complexes, these electron carriers rather, I would say, they are arranged in such a way that there is a zigzag pattern in that redox potential. So accordingly we will study. So we will start with the PS2. So it's not like that your PS2 cannot absorb, your PS1 cannot absorb the light simultaneously. They do, but that's how we will study. So once it will absorb the light, reaction center will also absorb it, right? Light harvesting complex will also absorb it. So light harvesting complex, after absorbing, pass it to the reaction center. Why? What will happen? You know that when they will absorb the light, what will they do? It will get excited. It will get Excited, yes or no? So excited one, because it is, so this one will be the excited because it has absorbed the light energy. We know that it is in the thylakoid membrane, or meanwhile here it is towards the lumen side, it is also having OEC. I'll come to this part later. So if it is getting excited, ultimately in this complete process, you people will just talk about, in this complete process, you people will just talk about oxidation reduction. Oxidation reduction. Dekho, oxidation, loss of electron, oxidation, loss of electron, reduction, gain of electron. If let's suppose this is a particular compound having high redox potential, this one is less. So high redox potential means it is accepting the electron from the one which is lower. So it is what? It is having the ability to oxidize other compound. है ना? Right? It is having the ability. It's a good oxidizing agent because it is getting reduced. Right? It is good oxidizing agent. Matlab it can oxidize the another one. It can ask them to lose their electrons. So that's how the things are connected. Is that clear? That's how the things are connected. Is that clear? So now they will pass it to the pheophytin. Now you might be thinking the diagrams that you have added, ma'am, pheophytin was not written anywhere. But a pheophytin na somewhere with this PS2 only. But remember, pheophytin is the primary electron acceptor. And here there is one more catch because this is uphill transport. So I will come to this later, but I think I should write it so that you should not forget. Okay, uphill. Then this is pheophytin. So pheophytin is believed to be the primary electron acceptor. Pheophytin is what? It is the primary electron acceptor, or I would say it is the the first one electron acceptor here. So it is associated with PS2 itself. So it got excited, right? Its electron, they have absorbed the energy, they will come to the excited state. You know that ultimately if there is the excited state, they have to come to the ground state as well, right? It's not like that if something is in the excited state, it can stay there always. No, it's mandatory to be on the ground state as well. So oxidation reduction will be there. So if it has passed its electron, it is oxidized, this one is reduced. Now it will pass it to, you know that which one is the next one? Plastoquinone. Plastoquinone, that is PQ, right? Once it accepts the electron, now I'm not discussing 1 electron, 2 electron, nothing at all. I'm not talking about that. It will become plastohydroquinone, and you can also call it as plastoquinol. And what's the catch here? The catch here is that this plastoquinone is not just an electron acceptor, but also a proton carrier. It is not something कि I will not just accept the electron only. If you think कि you will pass me the electron and you will say कि yaar, you are reduced now and now you are oxidized, na, right? Not at all, not at all, right? It is like I will also accept the proton. So now from where it will get the proton? Now to I'm showing you it as per that redox wala thing na. Just up to this uphill, then downhill transport. Uphill matlab this part I will elaborate later beta. So I believe this part is clear, hai na? I'm making these notes, but this portion I will elaborate later. So P60 se gaya pheophytin, plastoquinone. So I told you here that it is not just an electron acceptor, electron carrier, it is also the hydrogen acceptor. ठीक है? Chalo, whatsoever it is, done. So from where will it pick? Because it is in thylakoid lumen, obviously it will pick it up from the surrounding. है ना? From the surrounding. So yeah, it will also accept proton H positive from stroma and will become PQH2, right? It will become, so plastoquinone will accept the protons from stroma. Because it is not just the electron carrier, it is also the, right? Because it is not just the electron carrier, it is also the proton acceptor, a proton carrier. So that's why it will become like that. So in between these two, there is a Q cycle. So Q cycle is not the part of our syllabus. You know, when they accept one electron, then pass it further, then further help in their, you know, or this regeneration. So that is not the part of your syllabus. Whatsoever I'm telling you, you just stick to that. That will be more than sufficient. Now, this one will pass further the electrons to cytochrome B6 F complex. Done, beta. And then from that cytochrome B6F complex, it will pass to plastocyanin. Jैसे here you have cytochrome B, cytochrome F, plastocyanin PC, which is having the which is copper containing, right? Which is what? Which is copper containing. Which is copper containing, plastocyanin. So ultimately oxidation reduction is going on. Now here before, so I believe this part is visible to everybody. Hai na? Now I'll ask you one thing. If this further, like moving further, I would like to explain this first. You tell me one thing, if it is a PS2, if it is a photosystem 2, it is getting the sunlight, right? It is absorbing that. Right, it is in its excited state, it is passing its electron further. Now, if it is passing its electron further, matlab it will get oxidized, but it has to come to the ground state as well, na? Right? It has to come to the ground state as well, na? Then how will it come to the ground state? How? This OEC. What is this OEC? OEC means oxygen evolving complex. What's that, people? That is your oxygen evolving complex. Oxygen evolving complex. Here what will happen? So here this is obviously towards lumen side, है ना? It is towards lumen side. Here the photolysis of water, it's a very important point. Photolysis of water occur. I told you that in the photosynthesis the breakdown of water is there, and that is going to be here. That is going to be here. So the photolysis of water will occur here. Is that clear? Is that clear? So it is the photolysis of water that will occur here. So you will have 2H2O, right? So this oxygen evolving complex is having an enzyme, water oxidase. It is having an enzyme, water oxidase. And here there is a trick for all of you. You people can remember, that is Man Ka College. Man Ka College. You have Mn2 plus manganese, calcium and chloride. This is what we need for this reaction. So in this oxygen evolving complex, there is the breakdown of water in the presence of light. So you will have electrons plus oxygen. So this is the step where oxygen will release. Am I clear? This is the step where oxygen will release, and this is the step where stroma will have the protons, and these are what you need for this photolysis of water. So tell me if there is any doubt here, guys. Quick. Tell me if there is any doubt here. Quick. Sure? So, because these electrons, they will pass to the photosystem too. So, it was in the oxidized state. It will come to the ground state. Protons will be there in the stroma. I told you what is our agenda. We have to maintain the proton gradient. So, protons are coming here. Now, if plastoquinone will also accept the H positive from the stroma, it is also decreasing the H positive in the stroma, protons in the stroma. And this is how the oxygen will release. Now, this plastocyanin will pass the electrons to PS1. Now you know that it's not like that PS1 is not absorbing the sunlight individually. It is also absorbing it, beta. But I have to tell you the electron flow as well. So it will pass it to the unknown electron acceptor A. Unknown electron acceptor A. There are many further steps. And finally, it will pass it to ferrodoxin. And now you know that ferrodoxin is on stroma side. It is having FNR with it. What is FNR? Let me write it here. So your FD is ferrodoxin, which will ultimately, it is also having iron sulfur. FNR is ferredoxin NADP reductase. And ultimately, right, so they are towards stroma side. Their location is towards stroma side. That's what I'm telling you. So this, as the name is saying, ferredoxin NADP reductase. So beta, stroma mein you have oxidized NADP. So ultimately electrons will pass to this NADP. It will pick up the protons from the surrounding and you will have NADPH2. That's what we are going to have here. That's what we are going to have here. This is the whole process. This is the complete process that you have. I hope it is clearly visible to all of you. So PS1 will pass its electron. 2 passes its electron to pheophytin, which is primary electron acceptor, and this transfer is the uphill transfer. Why uphill? Uphill to not possible, but here it is possible because sunlight is also there, right? Sunlight is also there, Ananya. That sunlight is also there. So that's why this extra energy is. So that's why PS2 can provide the electrons to uphill, to the pheophytin, and that too against this redox potential. PS2 is having high redox potential. Pheophytin is having less redox potential. Still it is getting that because sunlight is also providing the energy. That's what you people need to remember. Now next point is rest. Right, so PS2 passes its electron to pheophytin uphill. Rest all is downhill transport. That's a direct PYQ beta. Rest all is downhill transport. Okay, now electron flows from PS2 to PS1. Again direct pointer. Next, oxygen evolving complex, where what will happen? Photolysis of water bring PS2 to ground state. Matlab it was an excited state, that one will bring it to the ground state. And plastocyanin bring PS1 to the ground state. And other points that you have to remember, which I have repeated, I guess, multiple times, that how the electrons will flow. Is that clear? Done, beta? That's the complete flow. This is the Z scheme, okay, and this is the non-cyclic photophosphorylation. So by this particular process, just say here, photolysis of water is also adding H positive in the lumen. When plastoquinone is accepting the H positive from stroma, it is again decreasing the H positive in stroma. When NADP oxidize, when it is getting reduced, it is also using the H positive from stroma. So because of this complete process, H positive will be more in lumen, that is pH will be low. H positive will be less in stroma, that is pH will be high. That will be done. So this proton gradient helps in ATP synthesis that we will understand in chemiosmotic hypothesis. Okay, done? Done, beta. So that's what you have to keep in your mind. And when it comes to the non-cyclic photophosphorylation, that is going to be a cakewalk. And even whatsoever I have explained, I have added here in the format of points as well. So if you really need to understand what non-cyclic cyclic photophosphorylation is, so here only ATP forms. It occurs occasionally. These are the bullet points here. Only PS1 involved, no FNR, occurs in stroma thylakoid. That's what you have. That's what you have. Occurs in stroma thylakoid. Done? Any doubts? So yeah, this is the cyclic phosphorylation. So here you people can see it will start from PS1. It will get excited, passes its electron to FRS. Full forms are written here as well. Ferredoxin, a reducing substance. Then to the ferredoxin. So ferredoxin will give the electron to cytochrome B6 because it is B6 and cytochrome F complex. So from cytochrome B6, it will pass to plastoquinone, plastohydroquinone, and it will pass its electron to the cytochrome F. So yes, it is of point when plastoquinone passes its electrons to the plastoquinone accepts electron, pick up the H positive from the stroma, and when it passes its electron further to the cytochrome B6F complex, it then at that time that H positive will be passed in the lumen, and then cytochrome F, finally PC, and it will bring it back. So this is the cyclic photophosphorylation. That pointers are important. Make sure you take the screenshot. Now this is chemiosmotic hypothesis given by Peter Michel, right? He was the one who gave this chemiosmotic hypothesis. So you can see this arrangement whatsoever I taught you. Just say H positive, it is accepting, it is passing, when it is passing the electrons to cytochrome B6F complex, the H positives are passed in the within the lumen, and then plastocyanin, and finally this. So whatsoever I taught you, it is there. Now, why do we need more H positive here? It's very simple, beta. It's very simple. Just say here also you have this ATP synthase. This, I'll mark it here in the book itself. CF0, CF1. Okay, you have CF0 and you have CF1. CF0 and CF1. So CF0 is within thylakoid membrane. Position needs to be very clear. Within thylakoid membrane. CF1 is in stroma, and here you people can clearly see. So this proton accumulation inside the lumen is providing the energy. Conformational changes will occur. So when these H positive, right, because here inside the lumen is high electrochemical proton potential gradient, outside in the stroma it is low. So sometimes in the diagrams they can even change that. Instead of high they can mention low here, here they can mention high. Then it is going to be a mistake. Are you getting my point? Then again it is going to be a mistake. So that's what you have to understand that, right? If you will make mistake here, so high to low, that's how it will move, provide the energy, ATP will form, beta. This is a chemiosmotic hypothesis where the high H positive, low is there. You have to be very clear about it. This is the complete process of ATP synthesis. So I told you in the light reaction where the ATP will form, where the oxygen will release, where the NADPH will form, that will be there in the stroma itself. So now I am open for the doubt, so you can freely ask me. Quick. Hanji. Quick. Quick, everyone. Hanji, quick. Sure. So that is it. So those who are learning it for right, that is what you have to understand. Yes. So those who are learning it for the first time, tell me if they have any confusion, if they have any doubt here. Quick. Quick, quick, quick. Anybody having any doubt? So I told you this is going to be the easiest topic. That's what you have to remember. So you can see the requirement for the chemiosmosis. You can see the differences also. So yeah, key difference from mitochondria and chloroplast is in chloroplast within the lumen you increase the H positive concentration, so within lumen pH is low. When you understand the respiration, so in respiration here inside the intermembrane space you try to get more H positive, and that's how this process varies. So this is what you have to remember. So any doubt, then do let me know. Chaliye. So next part is what? Next part is basically the dark reaction, which is a biosynthetic phase, right? Which is even the light independent reaction, Blackman reaction. I told you the word dark is misnomer here, and here you need NADPH2, right? You need NADPH2, you need the ATP here, and then here in this particular process, the carbon dioxide reduction will occur, which will give us the sugar. So C3 pathway, C4 pathway, CAM pathway is there. C3 pathway is beta your Calvin cycle. Right. Why it is C3? Because here the first stable compound that we will get, the first stable compound is your 3-PGA, that is your 3-phosphoglyceric acid, and we can also call it as phosphoglycerate, right? Right, 3 carbon containing compound it is. So in your C3 cycle, this is the gist beta. Which one is first acceptor? Initially it was thought that it is going to be a 2 carbon, but nahi. First acceptor of CO2 is 5 carbon containing compound. 5 carbon containing compound, that is your RUBP. So what is RUBP here? It is going to be ribulose 1,5-bisphosphate. So beta, because here I have used ribul, ul means it is a ketose, it's a 5 carbon containing compound, and it is, why is it the bisphosphate, why not the diphosphate? So in case of diphosphate beta, to 1 carbon both the phosphates will be attached. So when it is bis, so it is clear. They say 1, 2, 3, 4 and 5. 1 here, 1 here. So that's how it is. That is how it is. This is what we have to remember. Okay. Okay. That's the important point here. That's the important thing here. So reaction or C4 pathway may first stable. Why it is C4? Because here the first stable product is your 4 carbon containing oxaloacetic acid or oxaloacetate. So this is from where the question can come, right? So experiment beta, you can simply read that out. So I will clearly come to this particular part. So where this process will occur, this will occur in the stroma. So what's going to happen? Ribulose 1,5-bisphosphate, 5 carbon containing. So this one will accept your CO2. Right? So there are, because it is the C3 cycle, right? Calvin cycle, because Calvin and Benson, they experimented, they got it, right? They experimented in chlorella. You can read that experiment, very simple. So C3 cycle, because beta it is a cyclic process. So here first is reduction, first is carboxylation. First is carboxylation. Then there is reduction. And then there is regeneration. Regeneration. These are the 3 steps that you have here. Carboxylation, reduction, regeneration. These are the 3 steps that you have. Carboxylation, reduction, regeneration. So for 1 CO2 when you are fixing, this is CO2 plus water, carboxylation will occur. Beta, this will be catalyzed by Rubisco. A key step here. So what's that? Ribulose 1,5-bisphosphate carboxylase oxygenase. This is the name of the enzyme. This step is catalyzed by this particular enzyme. What is the speciality of this enzyme? It is the most abundant enzyme, beta, and it is the most abundant enzyme of biosphere. And this enzyme can do both carboxylation as well as oxygenation. Both the things can be done by this. Carboxylation as well as oxygenation, right? Both can be done by this. So, this enzyme mainly go for carboxylation, but if relative concentration of CO2, like let's suppose if CO2 is less than oxygen, then it can, this is a drawback, beta, it can do oxygenation and can cause and C2 cycle occur, which is also known as photorespiration, right? So because of this particular enzyme, actually photorespiration or C2 cycle occur, which is a wasteful process, which will not help in the assimilation of carbon dioxide, but rather will waste the assimilated CO2. So it is a wasteful process where ATP is used, no profit, only the loss, loss and loss. So this is the ribisco ribulose 1,5-bisphosphate carboxylase oxygenase enzyme, most abundant enzyme of biosphere, can do both carboxylation and oxygenation, but yes, it will go for carboxylation. But if the relative concentration of carboxylation is carbon dioxide is less, it will go for oxygenation, and then it will cause the wasteful process. So yeah, 3 PGA will form because 5 carbon, total 6 carbon. Then beta reduction is there. So in reduction, 2 ATP, 2 NADPH2 are used. That's what you have to remember. And then obviously they will again enter in the system, again will form. Triose phosphate will form. They will further form sucrose or starch. Why sucrose? It is the invert sugar. So in your phloem, it is non-reducing sugar. So in the phloem, the sugar is translocated in the form of sucrose. So sucrose is nothing but beta, it is a disaccharide. So sucrose is nothing but it is a disaccharide. So this disaccharide having what? Having, it is made up of glucose plus fructose. So this disaccharide, my dear lovely students, is actually translocated there. Then after this, you people will see when there is a regeneration, 1 ATP is again used. So that's why ATP and NADPH are required for it. And this is what you have to remember here. Any doubts? Done? Done? Sure? Okay. So now see, for one glucose molecule, because it is C6H12O6, we need to fix 6 carbon dioxide. So 6 turns of C3 cycle occur. How many turns? 6 turns of C3 cycle occur. So if in one turn, total 3 ATP, 2 NADPH used. So for one glucose, you know that 18 ATP, 12 NADPH2 are used. That's what you have to remember. So if somebody will ask you for 120 glucose, how many ATP are used? So don't confuse it, beta, because for one glucose, you are using 18 ATP. 18 ATP when you consider all the 3 steps. So for one, how many ATP are used for 120 glucose? That will be your answer. Noted? Noted? Done, beta? Done? So this is one type of MCQ that can come. This is one type of MCQ. Now next is, the second type is for synthesis of 1 या 3 glucose molecule, this is one you have to answer, 3 glucose molecule. For synthesis of 3 glucose molecule, how many ATPs are used in regeneration step? Now answer it carefully. Regeneration step, that's your answer question. In regeneration step, quick everybody. Quick. हाँ जी. So this is what I'm expecting you to answer, and I believe you all know that. देखो ना, read the question. 3 glucose, and they have specified the step regeneration. So for 3 glucose you have to synthesize, right? So you know that for 1 glucose, how many total steps of Calvin cycle will occur? Total 6 steps. So 6 steps, and in each step you have, in regeneration you just use 1 ATP. So 6 ATP here for 1 glucose. So 3 glucose are there. So accordingly you will have the ATP here, right? Right? So not for 1 Calvin cycle, I'm asking for 1 glucose, and for 1 glucose we need to fix 6 CO2, 6 turns of Calvin cycle will be there. So accordingly you have to answer. So this is the most important thing that you have to remember. Any doubts? Any doubts, guys? Any doubts? Though it is a marathon, I'm telling you each and every possible type of MCQ. I am telling you how you have to proceed. Right, beta? Right, beta? So please keep watching this channel. Keep supporting it. If you are new to it, you know that you have to hit the subscribe button as well. Am I clear? You have to hit the subscribe button as well. Am I clear? Sure? Chali, moving ahead. You have told you already about the Ribisco. Next is the C4 cycle. For that, you have to understand the Krebs anatomy. So C4 cycle is beta nothing but the hatch and slack pathway, right? So it is an adaptation. It is an adaptation of the plants, those who grow in the dry tropical regions where the temperature is very high. And this is the speciality of these C4 plants, that they do not experience, or it is very less photorespiration in their case. Why? Because there, and why it is the C4 cycle? Because here the first stable compound is oxaloacetate. Now what's the speciality here? This Krebs anatomy, right? So I would like to discuss that with you all. So we have to focus on the Krebs anatomy here. So this is going to be the last topic for this particular chapter, and then we will have a long break, and then we will start the rest of the portion, okay? So Krebs anatomy. Beta, the meaning of Krebs, do you know that? What is the meaning of Krebs? Krebs means wreath. What is this wreath, ma'am? This is the wreath. This is the wreath. This one, this is a wreath. Normally what is happening when you look at a C3 plant, देखो, C3 cycle will be there in all the plants, but there are special plants where C4 cycle will occur, but still they will also have the C3 cycle. मतलब in all plants you will see the C3 cycle for sure. Now here what's the point? When you look at the C3 plant, जैसे ये देखो, this is the vascular bundle that we have. This vascular bundle is covered with the bundle sheaths here, beta. है ना? And these are the surrounding mesophyll cells which are also having the chloroplast. Now when I look at the C4 plant, yes, I do have the vascular bundle here. I do have the bundle sheath cells here. But if you look at the surrounding mesophyll cells, don't you think that they are arranged like a wreath here? This is the first point that I have to focus. They are arranged like what? They are arranged like a wreath. Now, moreover, these here, we will see in C4 plants that the chloroplast show dimorphism. Yeah, dimorphic chloroplast is there. It is having two forms. Let me address that also. So first of all, let's suppose this is vascular bundle. These are the bundle sheath cells in the surrounding. And if these are the mesophyll cells here, right? If these are the mesophyll cells here, so you have to see one thing. And what is that one thing? that these bundle sheath cells, first of all, these bundle sheath cells, they are having thick walls and they are impervious to gaseous exchange, right? They are impervious to gaseous exchange. Is that clear? They are impervious to gaseous exchange. Plus, in bundle sheath, you will have large sized chloroplast. Here, rubisco present. There is one more enzyme that we understand in C4 cycle, that is pepcase. So pepcase means phosphoenol pyruvate carboxylase enzyme, dear students. Phosphoenol pyruvate carboxylase enzyme, dear students, that is absent. So here in bundle sheath cells, you will see C3 cycle occurs, but no C4 cycle. That's how in the C4 plant, C3 and C4 like cycle, they are separated by the space here. Now, in the mesophyll cells, right beta, small sized chloroplast. Bus. Now for this paper, this is what you have to remember. Small sized chloroplast, no rubisco. If no rubisco, then there is no chance of having C3 cycle. So no C3 cycle. Pepcase present. So pepcase, I already told you. Phosphoenol pyruvate carboxylase, this is present. So C4 cycle. So if here the question can come where the C4 cycle will occur, mesophyll cells. Where the C3 cycle will occur, bundle sheath cells. Large size chloroplast, small size chloroplast, but it is impervious to gaseous exchange. Now, ma'am, how is it beneficial? Very simple. Pepcase is not affected with, pepcase can fix CO2, or I would say even in low CO2 concentration. So, no photorespiration, which is a wasteful process. So, we won't see any photorespiration here. So that is going to happen. That's the beauty here. Are you getting my point? That's the beauty here. That's going to happen here. Is that clear? That's the beauty here. That's going to happen here. So in these cells, the carbon dioxide will be fixed. Then further we will pass the compounds to the bundle sheath cell, where again once decarboxylation and then again the carboxylation will occur and C3 cycle occur here. So even if there is the low CO2 concentration, because high temperature is there, CO2 availability is affected. So still, because here normally C3 cycle will occur, so no photorespiration will occur. So I will give you a wonderful diagram here. First of all, C3, C4 cycle. Have a look at the arrangement. So these notes will be provided to you in our WhatsApp group. Right, you have to join that. Everything, all the details are given in the description box. So look at this. This is the best way to revise it. Have a look. Phosphoenol pyruvate. So this is the mesophyll cell having what? Small sized chloroplast. Having what? Having small sized chloroplast. So you can see, so here this one is the first acceptor of CO2. So these flowcharts will make it very easy for you. First acceptor of CO2 it is beta. Phosphoenol pyruvate, CO2 will come. So this one is the first stable product. So this particular process is also known as cooperative photosynthesis. So first stable product is a 4-carbon containing compound. So here it is using NADPH. It is forming malate. That malate through plasmodesmatal connections will pass to the surrounding bundle sheath cell, and they are impervious to gaseous exchange. This is the plus point here. So this malate beta, this malate, here we have used NADPH. So here NADPH is resynthesized. So here we will not count it anywhere. Now here, because this is the step where decarboxylation will occur. Right. This is also a kind of question in C4 cycle. How many step decarboxylation या carboxylation will occur? Okay. So you people can see here NADPH2 has formed. Here NADPH has used. Here it has formed. Here decarboxylation will occur. Malate will be converted to pyruvate. This CO2 will go in Calvin cycle. Normal Calvin cycle will occur. Pyruvate will go back to the mesophyll cells from bundle sheath cells. Why? Because it has to regenerate. It has to regenerate the phosphoenol pyruvate. So when you regenerate, right, PEP from this, so 2 ATP are used. So this is the extra expense that you pay. So for one glucose in C4 cycle, right, you will see 12 ATP extra are used, and overall because C3 signing on, so same 18 ATP and 12 NADPH will be used. So in C4 cycle total 30 ATP and total 12 NADPH will be used. So this is what we have to keep in our mind. Is that clear? So this is the extra expense that we pay, right? Extra 12 ATP. Why? Because in one step of C4 cycle, right? One step of C4 cycle, 2 ATP are used for regeneration. And you know that here also we will overall take 6 turns. So this is all about the photosynthesis. This is done and dusted. I hope you are able to remember that. If not, do revise it. Now because we didn't take any break, like the break was for the 15 minutes only, and we are almost about to complete 5 hours. So this is also the tables that I have added, right beta? So as of now for this NEET 2026, stick to these topics, do revise this particular diagram, and here you have ABCD points as well. So the next part that we have to study is the respiration in plants. That let's understand after a long break, not the short break. Let's have a break from 4 to 4:45. What say? It's a long break. You can also take the rest and then we will discuss it further. Done? Okay. Sure. So see you all after 45 minutes. Enjoy this long break. Then we'll see you all. Okay.